So good morning to you all. Welcome to the second day of the NORA online conference. I have the privilege to open this day. My name is Karel van den Wijngaard, representing ARC Nature from the Netherlands. And I'm coming from The Hague, close to the beach here. I'm really glad we do have this online opportunity to connect and to share. So thanks again for the people who did the hard work to make this possible. Amongst others, Philine, Katrien and Boos especially, and all the others involved. And at the same time, just stating the obvious, I'm also sad that we cannot meet live yet. Let's stick with the yet. Let's hope that next year it will be possible. But nevertheless, we have lots to talk about. So the program today is quite fully packed. Today is built around 4 main sessions. First of all, there will be a main session on production, then one on management, a third one on monitoring, and the last one on the outputs from the NORA working groups. And in between, there are other possibilities and options. There will be a presentation of Edo Knechtering, representative of the Dutch government, on how to ensure Bonamia-free introductions. There will also be the poster sessions. I believe there will be 3 moments during the program where you can visit the poster room and where the people who made the posters will be present to talk to you. And the whole day, just like yesterday, there will be, of course, the opportunity to mingle and to use all the options that Gather Town offers. And at the end of the day, at 1700, there will be a happy hour even. I'm not sure what it means in a digital environment. I guess it means bring your own, but let's see. Before we really dive in, I'd like to share just a couple of thoughts briefly, because I was triggered by some of the things that Betsy Peabody mentioned yesterday. So of course, during this conference, there is a lot of focus on science, on research, on data, and it makes sense because it is necessary for us to do our work, to know what to do, or to build understanding, as Betsy put it yesterday. But that's all about the what. And that's probably not the reason why we do all this. It's not about the why, at least not for ARC and not for me. So I suppose the real motivators are more about feeling and believing. For me, or for ARC, it's about feeling the necessity to change our way of interacting with the marine world. So the necessity of giving something back instead of just taking, as we have done for too long now, for this, let's say, past century. And I suppose it's about believing in shellfish reef restoration as an active nudge for marine nature, as a kind of positive intervention. And of course, for NORA, the focus is on oysters, on shellfish restoration, but ARC is also interested in the question, what other interventions are possible? So perhaps starting with shellfish, perhaps other shellfish species like horse mussels, for instance. Actually, at the moment, there is a first very small scale experiment running at the Dogger Bank, initiated this year by Emily from WWF, the Netherlands. But perhaps also seagrass, which was also mentioned by Luc Helmer yesterday. And another thought is how to use those observations that some of us did on the eels while working on shellfish restoration. And I guess I read something in the chat yesterday that Joe Preston will be talking about it a little bit today. Or even a pilot that we haven't imagined yet. In fact, ARC has been thinking and talking internally for this last year about rewilding at sea. And hopefully at the next time, if it fits within the scope of NORA, of course, I might be able to share that story with you if it's finished. But for the moment, of course, our main example is oyster reef restoration. And in fact, ARC is literally working on it today, placing oyster cradles with natural collected spat in the Voor Delta. As a first, at least for us. So if I try to summarize, I guess I would say, let's not just share our research projects and results, let's also share our stories or our other stories. So that's all from me for now. Let's enjoy this second day of the NORA conference and let's dive in. Next up is the production session, the first main session of today.
Okay, thank you, Karel. So we, Pauline and we are pleased to welcome you at the oyster production session this morning. The session is composed of 5 talks, followed by 20 minutes dedicated to questions, comments, and interesting discussion, I'm sure. I take the opportunity to remind you about the posters and projects to be seen in the other rooms of Gather Town. And for now, I wish you a nice session.
Thank you, Bérenger. So it is my pleasure to introduce the first speaker, which is Bérenger. Bérenger is an early career scientist with experience and knowledge on hatchery techniques. He is the hatchery manager of the Helgoland Oyster Hatchery, and he conducted his PhD within the German flat oyster restoration project, Restore at the Alfred Wegener Institute. Please go ahead.
Thank you, Pauline, for the nice introduction. I'm delighted to present you today the Helgoland Oyster Hatchery. The hatchery is located on the beautiful island of Helgoland. And where is Helgoland? Here on the map of the different seed production sites in Europe, the hatchery is represented by the star. For those who don't know the island already, the island is about 1.7 square meter in the German North Sea and is about 29 miles away from the mainland. Here is our team in pictures, excluding me and Bernadette. From left to the right and top to the bottom is Lennart, Clemens, Jan, Aldi, Corinna, and Tao. To be noted that Jan and Tao were our fantastic master students of 2021, and that my fantastic colleague Corinna is working on the outreach of the project, so not directly related to the production. My other colleagues are fantastic as well. That's it for the introduction for today. If you want more information of our contact details and our contact details, here are 3 links. And above all, you are more than welcome to visit us on Helgoland. Now the design. Before any construction, we thought necessary to make a state-of-the-art in order to understand or optimize the production techniques as much as possible. We started by visiting many hatcheries in Europe, mainly Ostrea edulis, but not only, and also hatcheries in Australia and New Zealand. Here are 4 pictures of hatcheries as example: Roomwannerseke in the Netherlands, Spat-on-shell in New Zealand, Fermarine d'Ildara in France, and the Ifremer Argenton in France as well. In the second step, we got to know experts and hatchery enthusiasts, and many of them are members of NORA today. As a good example, we have established a cooperation with the CRC Bretagne Nord in France, and we are looking to develop it further and developing more cooperations. On the third step, we have, and this is also an example of cooperation within NORA, try to establish a state-of-the-art in Ostrea edulis seed production, going through the techniques and biological data found in the literature, and this by producing a review paper on the subject. Here it is. And on this last stage, I will elaborate it a little. The review paper covers the whole range of Ostrea edulis seed production techniques, including hatchery, sea-based collection, breed pods and breeding ponds, but also the biology of the species, stresses, the short history of production technologies, and the genetics applied to Ostrea edulis. It goes without saying that for the design of the hatchery, the sections developed on the biology and stressors of the species were particularly of importance. Just as an example, the biological data corresponding to the understanding of the life cycle, including, for example, sex ratio, fertility rate, those were essential elements for the dimensioning of the breeding units. Data such as the start or the suspicion of biological reset at around 7 or 10 degrees Celsius, data on larval size and life cycle phase duration were also essential for estimating and or planning breeding operations and tools. As an example, the size of sieves, the mesh size of sieves. Here are 2 Other data table as example, a list of pathogens and parasites found in the literature, including here 6 Vibrio species we were talking yesterday, and the list of known pollutants affecting Ostrea edulis larvae. To be noted that only 3 heavy metals are known. That's it for the review in this talk, and for more infos, please visit the DOI number. Finally, it is very important to emphasize that the design of the hatchery was, despite many adaptations, inspired by the more general manuals that everyone here should be or are familiar with, Dupuy et al. 1977 and Helm et al. 2004. And many others, but there would be not enough time to mention them. And yes, I like to read. The hatchery today. This is what the hatchery looks like from a schematic point of view, and they are split in 8 different parts. Seawater filtration and sterilization, food production on small scale, medium scale, and large scale, quarantine of new broodstock, conditioning of broodstock, larval rearing, larval settlement, nursery, but also laboratories, storage, offices. Water. Here you can see our water treatment equipment for the entire hatchery. The autoclave is exclusively reserved for small volume cultures, a small volume of microalgae cultures. For the rest of the rearing units, the water is successively filtered through first a cyclone filter for sand, back filters down to 1 micrometer, and then sterilized by UV lamp. The food. Food production is based exclusively on the production of microalgae. We cultivate 4 species, and they are Chaetoceros mülleri, Rhodomonas salina, Isochrysis nuda, and Tetraselmis susica. The culture starts from the strains in incubators on the pictures above left. Then in small volumes, 100 milliliters, 500 milliliters, 1 liter, and 5 liters, depending on the species. Then they are transferred to what we call in the intermediate cultures in 20 liters in carboys, which are the 2 pictures on the right. After the carboy stage, the microalgae are grown in large volume of about 500 liters cylinders, and the hatchery has 36 cylinders. The microalgae are then harvested and distributed to the different units of the breeding. As for the new broodstock arriving, as shown here, well, they are cleaned. The oysters are cleaned and scrapped to remove as much of the fouling as possible, and then they are bleached, rinsed, and placed aside in a tank quarantine for at least a month before being prepared for conditioning. Preparation of conditioning, then the preparation is divided in 3 steps. The first step is to identify the oysters. So we do glue sheep ear tags on the upper valve. The second step is to take their biometry of each oysters and their weight. The third step is to establish the dry weight of a representative sample of the broodstock. Here are our conditioning units with an example of 9 tanks with their outlet leading to a collection tank for future larval collection. And we have 2 broodstock origin for 2021 is a Norwegian and a Scottish broodstock. At swarming, the larvae are concentrated in collectors or sieves. They are then counted, mortality rate is estimated, swimming behavior rated, etc. The larva rearing is a 36-time flow-through system, around 5 liters cylinders each, as described by Ricardo González Araya in his article and the thesis. For the settlement phase, here are our structures. They are 18 times flow-through tanks of 500 liters each, with 3 times onion bags filled with 25 kilos of Ostrea edulis shells inside. The shells are, of course, washed, salted, and sterilized before being put in the water, which is 15 days before settlement for the development of the biofilm. The first results of this talk would be the success of our conditioning method and the production of larvae in 2021. For this, we have established a conditioning protocol by, in fact, collecting all the findings of our colleagues so far and found in the literature as optimal photoperiod, optimal temperature rise and fall, and optimal microalgae composition and diet. And this we applied to 90 broodstock oysters of each origin. Although we are still in the process of interpreting the results, we can already point out that the production of larvae were possible for both origins with the conditioning method established. This year, a total of 32.4 million larvae were produced, and we are looking forward to repeat this experience next year again. Thank you for your time, and I will be happy to develop to discuss further with those who are interested. Thank you.
Oh, thank you very much, Bérenger. For some reason, I can't start my video. But thank you very much for this interesting talk. And we will move on directly to the next talk. So if you have questions, please save them for the question and answer session after the presentations. So the next presenter is Nicolas Araujo Piñeiro. He's a oceanographer with a background in aquaculture, and he recently graduated from the Aquatic Science and Technology Master Program from the Technical University of Denmark, and he focused on low trophic aquaculture and restoration. Please go ahead, Nicolas.
Okay. Hello to everybody. Yes, my name is Nicolas Araujo Piñeiro, and this is my first time of NORA conference, and I'm really happy to be here and contribute to this great community. I'm going to share with you the study that we did for a master thesis last year, and which was supervised by Camille Bertolini. First, I would like to start by talking of the context of our experiment, starting by two of the main reasons that limited the success of Ostrea edulis in the environment, which is the lack of adequate substrates and not having enough larvae, and then the materials that we should use for restoration projects. A big emphasis is put on nature-based materials. That's why we chose to work with spat-on-shells. And then regarding the treatments that we evaluated, we focused on two of the reasons that affect the larva preference in terms of settlement, orientation and site, and the presence of conspecifics. And then I would like also to mention the scale of the project. We've seen a restoration project where big volumes of cells are put into the environment for creating this good, adequate substrate. And then on the other side, we have a small experiments under really controlled conditions, like in laboratory. And in our case, we wanted to provide knowledge that could be used for research or commercial hatcheries. That means that we use the material and protocols that they are currently being used in the Donnan Shellfish Center. So the first experiment that we realized was comparing the first treatment, the shells method for spat-on shells, and the traditional method, which is powder made of Wijngaard shells, as you can see in the picture. Then the second treatment, we compared the two species of shells, Ostrea Wijngaard and Ostrea edulis. Then we also compared the orientation, how does affect having the shell facing up or facing down. And then we also compared the difference in larva settlement rate between the inside and the outside of the valve. Here is a diagram that you can see all the treatments. We have 3 tanks for the traditional method, another 3 tanks for Ostrea Wijngaard shells, and another 3 tanks for Ostrea edulis. And within each of the shells treatment or shells tanks, we have the other treatments, the side and the orientation one. So this is actually the look of the experiment. That's what the experiment was carried out. And this is exactly the same section used for traditional and production of shells, of spats, excuse me, in the Donnan Shellfish Center. So we use the exactly same system. In this slide, I want to guide you through the steps that I follow for realizing the experiment. First, the collection of the shells. The zygotes, I collect them myself from the shore, whereas the zygotes, yes, I collect them myself. Then the edulis, they were fished with a dredge in the Limfjorden, and both were kept outside to be exposed to rain and sunlight to get rid of any potential disease. And then I keep them in a bath 24 hours and I clean them very thoroughly to remove any particle or material that could have attached to them. Then I dry them out and I put them for 24 hours in the tanks before adding the larvae. And when I did, I put an average of 30,000 larvae in each of the tanks. And after 8 days, we checked the settlement. As you can see in the picture, we had the spats attached to them. But I realized that it was not that easy to count them from the outside because of the color, and they were very small. So I decided to leave them more time for them to grow and avoid miscounting of the numbers. So the results that we found is that the powder, the traditional method, was more successful than the shells, as you can see in the picture, and the settlement of the powder was within the range that they used to have in the Danish Shellfish Center. Between the two shell species, we didn't see really a big difference. They even prefer zygotes. And for the orientation, also very similar results. And for the inside, outside, we saw a little bit preference for the outside side of the valve. And when we run the statistics, the only two treatments that they show statistical significance difference were the comparison between the traditional method with shells and the inside and outside. So then we carry out the second experiment. And I would like to stop here a little bit to explain why we did this experiment. In the first experiment, we analyzed the quality of which substrate to use and how to use it. But then thinking about upscaling, we had the question whether the larvae are capable to swim inside the tank when it's filled with water and larvae. So we designed this experiment. And what we did is, like, in 90-liter tanks, we fill them with water, shells, and food. And then in the first treatment, we put all the larvae, once the tank was filled with the larvae and shells, altogether 96,000 larvae. And in the second treatment, we did it by steps. We fill a layer of shells and water, then we add some larvae, and then we repeat that 5 times. So what we wanted to evaluate is, like, if the larvae can distribute evenly in the water column, if they can swim between the cells through the whole water column. And we also monitor the food concentration, the oxygen, and the temperature in each of the layers in both tanks to be sure that they were not affecting the results. And we keep them 63 hours in these tanks with no exchange water. So these were really extreme conditions for the larvae. And as you can see in this picture, this is the look of the experiment. And the results that we found is what we expected. In the top treatment is the one that we put all the larvae together. We saw a difference between layers, and in the layers treatment, we didn't see any difference. And the mortality that we saw were really high. We were not surprised by that. We were expecting high mortalities. And in the graph, you can see the difference between the both treatments. It was very clear that the treatment where we put all the larvae together, they remain close to the surface. So the conclusions of our both experiments is that Ostrea zygote shells are as efficient as edulis for spat-on-shell production, which is good news in terms of cells availability, because zygotes is more available than edulis. And then related to the second experiment, we could check and be sure that high-tech equipment and hatchery conditions are not essential for upscaling and producing big volumes of a spat-on-shell, which allow us to maybe produce spat-on-shells big volumes in different places without these requirements. And lastly, I would like to say that the Danish Shellfish Center recently opened a new hatchery, meaning that they are really ready for upscaling the spat-on-shell production, and they are currently working in a Bonamia-free certification for producing a spat-on-shell Bonamia-free. And that's all for me. If you have any questions, you can reach me in that email. And I would like to say thank you to all of you for listening, and also to Pascal and Bernie for their help and guidance in this process. Thank you very much.
Thank you, Nicolas, for the nice experiment, and that's very interesting to me. Let's discuss afterwards, I would say. So now it's time to me to introduce Pauline. Pauline Kamermans is a marine ecologist with over 25 years experience in marine bivalve research. She has worked on optimizing provision of shellfish seeds for aquaculture through hatchery production and spat collection in the field. Currently, her focus is on native oyster restoration in the North Sea. Pauline, the floor is yours.
Thank you very much. So today I'm going to present some results of hatchery production of Bonamia-free and resilient flat oysters. And with resilient, we mean either resistant or tolerant. And I did this work together with my colleagues Ainhoa Blanco and Pim van Dalen from Wageningen Marine Research, and Mark Ermgassen from Wageningen Bioveterinary Research, in cooperation with the two hatcheries in the southwest of the Netherlands, Nienke Bakker from Roem van Ierseke and Marco Dubbeldam from Stichting Zeeschelp. And we also cooperated with Pascal Jacobs of the NIOS and with Manel Vera and Paulino Martinez from the University of Santiago de Compostela. And the research was funded by the Ministry of Agriculture. So why are we interested in hatchery production of flat oysters? Well, as you know, there are a number of restoration projects currently going on in the Dutch North Sea. They are indicated here by the stars. And at present, the flat oyster is virtually extinct in the North Sea. And the Berlin recommendations advise to use oysters from disease-free areas when you want to restore oysters in the area where it wasn't present anymore. Now, it's not so easy to get oysters from disease-free areas. As you can see on this map, this is the distribution of Bonamia. Bonamia is the main disease in the North Sea area. And these colors indicate whether it is an infected area or a disease-free area. And these are two locations where, at the moment where when the publication was made, it was not certain whether Bonamia was present or not. And for the restoration projects I showed on the previous slide, oysters were sourced from Norway and from Ireland. Now, if you take oysters from a disease-free area, they are probably susceptible to the disease. So if for some reason the disease would arrive at these restoration areas, then you might have mortality. So we need Bonamia-free and Bonamia-resilient oysters. And as you can see on this map, the area where the hatcheries are located is an infected area. So we developed a method to select Bonamia-free oysters from a broodstock that was collected in an infected area. So we would cut a piece of gill from the oyster and then send that to the bioveterinary lab, and there they would check each oyster whether it was infected or not. And in the meantime, the oysters would remain dry with a number. And here are some of the results, where you can see when they are free of Bonamia, they are green. And then they can be used to produce spat, larvae and spat. And we checked the larvae and the spat as well, and they were Bonamia free. So the method seemed to work to produce Bonamia free oysters, but are these also resilient? And for that, we worked together with the University of Santiago de Compostela, and they checked the presence of resilience markers. So we had the Bonamia-free oysters from the Bonamia-free broodstock, and we had Bonamia-naive oysters from the Wadden Sea, where Bonamia had not been detected. And the analysis in Spain showed that there are more resilient markers in the Bonamia-free group compared to the Bonamia-naive group. Then the next step was to challenge these two groups of oysters. So we placed them in the Lake Grevelinge, and Lake Grevelinge is an area that has been infected with Bonamia for a long time, as you can see in this graph. So we placed the oysters in cages in the area, and we checked their survival. The survival is shown in these two graphs. We did it last year and this year. So we had the group that was produced by Roem van Ierseke, the Bonamia-free and resilient oysters, and the oysters that were produced at NIOS, which are the Bonamia-naive oysters. And both years we found that the survival of the Bonamia-free and resilient group was higher than the other group. And these two groups were also checked again for the presence of resilient markers. So on this graph, you see that was done twice this year. On the left, you see the Bonamia-free group, and on the right, you see the Bonamia-naive group. And there are more resilient individuals in the Bonamia-free group compared to the Bonamia-resilient group. And we also checked these oysters for the presence of Bonamia. And what we expected was that the Bonamia-free group would not have Bonamia infection, but that the Bonamia-naive group would have Bonamia infection. However, no Bonamia was detected in any of the individuals, despite the fact that there was more mortality in the Bonamia-naive group. So, in conclusion, I'm quite happy that we developed a method to screen the broodstock from a Bonamia area and to produce Bonamia-free oysters. And it is also very nice that the Bonamia-free oysters contain more resilient markers and show more survival than the naive individuals. However, the last piece of the puzzle, that the Bonamia infection is missing. So I'm not so happy about that. There was no Bonamia detected in Bonamia-free or in Bonamia-naive oysters. So that leaves us with some questions. Why was this not detected in the Bonamia, in any of the oysters? Was there no Bonamia present? We still need to find out more about that. Or why did the naive oysters show more mortality? Was there another reason why their survival was less? Or maybe there is another explanation that we don't know of yet. So that was my presentation. Thank you for your attention.
Thank you, Pauline. I have already questions. I keep it for later. I will definitely follow this research. So now it's time to welcome Dr. Nik Sachlikidis. And Nik is a scientist and manager who has worked in research in government and commercial aquaculture internationally over the past 2 decades. He is currently the managing director for aquaculture portfolio for the Cadman Capital Group, who is the owner of Orkney shellfish hatcheries. Nik, the floor is yours.
Hello, NORA Conference. My name is Nik Sachlikidis, and I'd like to thank you for having me here today on behalf of the Orkney Shellfish Hatchery to discuss the development of our screening techniques for Bonamia that we use in our hatchery. Firstly, a little bit about us. The Orkney Shellfish Hatchery was established in 2017 and was purpose-built as a state-of-the-art facility for the production of shellfish products. It's located in the very far northern reaches of Scotland in Orkney, and at the moment it currently cultivates native flat oysters and European clawed lobsters. Our mission is to be a leading producer of native oyster spat, and our goals really are the commercial production of high quality native oyster spat so that we can support restoration efforts both in the UK and internationally, and to provide a platform for the development of farming of native oysters. Our journey started in 2017 when we began the process of restoring an old and dilapidated oyster hatchery into a modern facility capable of producing high quality oyster spat. This took us the best part of 2018, and we had our first broodstock onto the site in 2019. In 2020, we began our research trial phase. We produced our first spat on the site at a production scale that was a research scale, where we refined our techniques and knowledge about native oyster production. The learnings from 2020 were pivotal to our 2021 season, where we undertook our first commercial growth phase. We scaled up our production to more meaningful quantities, and you can see from the pictures above, larger amounts of broodstock, larger larval capacity, and larger algae support. This year, we've achieved our commercial survival targets and our setting targets, and we have sold for the first time into the restocking market. We've also put our first animals out onto farm for grow-out trials through the summer. We're really excited for our 2022 season. We're currently undertaking our second commercial refit and upscale, where we will expand our capacity in our team over this next 3 months to produce significant quantities of native oyster stock. Along the way, we've met some hurdles to the commercial culture of native oysters. And the first one really was just being able to find and access broodstock animals from Bonamia-free areas. And that was really challenging and sort of highlights the plight of the animal, really. But beyond that, when we bring them into the hatchery environment, the bottleneck was our ability to screen all of the incoming broodstock in a non-destructive way that we could then have a very high degree of confidence that our animals were clean and were free of Bonamia, and we could produce from those with confidence that the seed stock would also be Bonamia free. The key difficulty, of course, was Bonamia. It needs no introduction within this forum, but it's important to mention here that it is a parasite and that infected animals don't always display clinical signs of the disease. It does affect animals that are a little bit older, so therefore broodstock size animals in our context, and the transmission can occur directly from oyster to oyster. So this allows for fast horizontal spread within, let's say, a contained broodstock holding facility. So it's very, very difficult to contain if it were to be in your hatchery. The current Bonamia testing methods are PCR analysis and a range of histology preparations. And the problem with that is that they're time consuming, they're destructive, so any animals that we do test actually die of the testing methods, and they require a high infection threshold before we can actually pick the infection up in the testing. So the need is for a fast, non-destructive method for screening broodstock. For this, we've turned to technology, and specifically eDNA technology. And this is built on the premise that all organisms release DNA into the water. We can filter that water and then test that by PCR for background organisms. And what that does is give us a presence absence for Bonamia in a non-destructive and highly accurate way. And we can also test multiple animals simultaneously or multiple populations simultaneously. Jorgensen 2020 published some work that was pivotal to us being able to achieve this in our broodstock quarantine process. So to explore the quarantine process a little bit more thoroughly, we should have a look here at a standard process that in the past has been used to try to produce Bonamia-free spat. And it starts with the collection of broodstock from Bonamia-free areas. The animals are then cleaned of pests and epiphytes, and they're held in a quarantine for a period where they're observed and looking for obvious physical signs of that before being used as broodstock within any hatchery. And the problem with that is there's no direct verification of the absence of Bonamia. It relies upon the source stock being cleaned. A best practice situation might be the same process as above, but with the addition of a PCR test or histopathology to determine the state of infection for those oysters or for a proportion of those oysters. And that creates a barrier to that hatchery for Bonamia. And the only problem, there's two problems with that, and that is that there's a loss of a percentage of broodstock in the testing process because it's lethal. And the other side of it is the remaining broodstock don't necessarily aren't necessarily verified free of Bonamia. It's a probability estimate based on a sample. Where we're using the eDNA technology in our process is that we are adding an eDNA step, and that is directly measuring the presence absence of Bonamia in the remaining creatures or the remaining animals and broodstock, and it provides another barrier to Bonamia for the hatchery. So there's another step in here where we actually can directly measure each and every animal that's coming into the hatchery before we start using it for breeding. So the broodstock quarantine methods for Orkney Shellfish Hatchery are that we source broodstock animals from known Bonamia-free areas. We put them in a purpose-built quarantine facility that's off-site from our main hatchery facility, go through the usual process of scrubbing and observation, and then send a proportion of those oysters to Marine Scotland for destructive testing. As that added step, we then keep them for a 24-hour period without any water exchange. We take water samples and filter that water and then send that to the lab for testing for traces of Bonamia DNA. These samples are then analysed in triplicate and under best practices. And they're compared against controls. And our controls for this are a positive real Bonamia DNA, a negative blank sample, an internal control, and a screening for native oysters, which just tells us that that filtration process has worked properly and that we're getting oyster DNA as well. And the validation for that has shown that the results are consistent and repeatable and they're accurate. From our batches that we've sent, our samples have come back free of Bonamia following a 4-week quarantine period, and then at which point they are moved into the hatchery environment. So this method allows us to detect Bonamia at a much earlier stage in the infection and at lower concentrations than the existing methods. But really what we're talking about is that latency period and how long do oysters take to get to, infected oysters take to get to a point where the detection limits can be reached. And therefore, what is the minimum quarantine time to develop that latent low-level infection? So we have used a 4-week period in our quarantine, which we feel is a very generous amount of time, but can we minimize that? Is it valid after a few days or a week? And that's an area that we need to understand a bit more deeply. And the other part is next steps to develop other tests for other pathogens. So the next steps from a hatchery point of view are, of course, our commercial scale up, our second phase. Continued search for disease-free broodstock populations where we can take some broodstock from those populations and add them to our hatchery library of broodstock geographical strains. And the idea is to cater to different restoration projects that may want their spat from different geographical areas. So we are already carrying several lines and we want to continue to add to that. And the goal is really to be able to provide singles and spat-on-shell to restoration projects and farms. Thank you for having us. If anything in this talk has been interesting to you, please don't hesitate to reach out to us on the contact details below. Have a wonderful conference.
Thank you very much, Nick, for an interesting presentation. We will now continue to the last presentation, and that is a presentation by Camilla Bertolini. She has a bachelor's degree in marine biology and coastal ecology from Plymouth University and a PhD in biology from the Queen's University in Belfast. After 2 years of postdoctoral research in the Netherlands Institute of Sea Research, she became a researcher at the Hofstede University of Venice, where she is now a Marie Curie Sklodowska Fellow. Please, Camilla, start your presentation.
Hello, everyone. I'm Camilla Bertolini. I'm from the University of Hofstede in Venezia, and today I will present you my Marie Curie project at MAREA, which stands for Matchmaking, Restoration, Ecology, and Aquaculture. And my supervisor is Professor Roberto Pastres. So what is the idea behind MAREA? Well, the idea behind MAREA is that of restorative aquaculture, which is merging of restoration and aquaculture. And this is meant to bring environmental benefits, but also economic benefits, and show you that the two concepts of environment and economy are not necessarily strictly separated, but can be linked. And this linkage will also aid to optimize the use of maritime space, obtaining both goals. Just MAREA in practice, what it is using areas which are already used as Mytilus cultivation, so concession area. Mytilus here is mostly cultivated in suspensions, in ropes. And the idea is using the space behind it, like underneath it, to restore Ostrea reef as a kind of mother reef, and then collect the spat, the settlement for aquaculture purposes, and then also to keep on sustaining some of the restoration. Some of the questions that this project will ask is, well, first of all, what is the success of restoration of the reef under the farms? What is the interactions between Mytilus and oysters? What are the ecosystem functioning that can be provided by these restored reefs? Where does the spat go? What are the best collectors for the spat? And can there be enough for culturing and also for restoration? And then what can happen in a climate change context? So extend this to the future. The approach that we will be taking is, first of all, an experimental approach. So we will have an experimental site in the Venice Lagoon. At this site, we will be seeding some oysters, following growth and health of these oysters, look at the ecosystem functioning and services with the diversity, but also looking at nutrient cycling and also having some spat collectors out. We will also have some sensors which will monitor environmental parameters and continuous. And we will also do then some model which will enable to scale up some of the processes and understand really the feasibility for aquaculture on a larger scale, and will enable us to do some long-term. The things that we did so far was a literature review. So this can be found online. identifying knowledge gaps for successful restorative aquaculture of Ostrea edulis. And what we did was a keyword analysis where we identified some key themes, and we had mainly papers that were concerned with diseases. We also did a manual search for restoration and for aquaculture papers, where we identified some of the subtopics within, and so shared some issues, and we found that there were some shared issues with larval production, settlement, and substratum between restoration and aquaculture. And we also looked at local historical knowledge, where we found that Ostrea was actually very highly abundant in the Venice lagoon and was started to decline due to unregulated overfishing and lack of sustainable, sufficient management actions to bring it back. And studies, once again, highlighted some of the issues with substratum limitation for successful settlement and the need for an ecological understanding of larval needs. So far, the experiment, we have our location in the southern lagoon of Venice, where most of the aquaculture is located. We have our site there in one of the mussel farms that looks like this. I don't know if you see the cursors, but we have the mussels are cultivated in suspension at the end of these orange ropes. And on the 23rd of September, we had some oysters arriving from Croatia, and I'm very helpful and thankful to the NORA partner that helped me source these oysters. So they arrived in a cool van, and we put them into these cages, mostly so that we could better access them. So we had around 2,000 oysters arriving, and these cages have trays, so different oysters and different levels of the trays, and then we will mix them up, so not always, so the bottom and top will be getting some, sometimes at the bottom and at the top, and then we deploy them at the mussel culture site with the help of the mussel farmers. So they are heavily involved in this project also, and they really like also being involved. I find great cooperation. Then, of course, we also have the lab part of the experiment where we are monitoring the growth of the different tissues. So we already monitored the time 0 and the time in the first month. So at the end of October, we already went out and everything looks still great. We're also looking at the diversity of the species on the shell. We'll be weighing the different compartments of the soft tissue, the reproductive tissue, and the shell. And this should enable us to then calibrate some of these models that we are doing. So the first model is, of course, a growth model, and we are taking the approach taken by Daniele Brigolin, which developed an individual-based, an IBM model based on the scope for growth. So here we have the food quality and quantity and the temperature that goes in as forcing, and of course this will then give you the energy available for growth, which is the anabolites, how much they've eaten, versus the catabolites, how much they expired, and so what's left over goes for this is the energy that is available for growing, and the growth will be separated into 3 compartments of the somatic tissue, the reproductive tissue, and also the shell. And so this is just an example of the somatic tissue growth. And so far, we're parameterizing this model for Ostrea. And another model that we're working with is that of larval dispersal. So we are starting to using the open drift module for Python. And this is based on the transport model. So we'll need a local hydrodynamic, but we can also add some behavioral characteristic of the larvae. So far, we just got acquainted with the larval fish module of the open drift. And we're searching also for Ostrea-specific parameters and waiting for actually the hydrodynamic model of the lagoon for the last few years. So we can try to estimate where we'll be next year, and then we'll know where to put the settlement plates. So in the next future, we keep on monitoring the growth so that we can calibrate these parameters. We use this model as an aid for modeling the fluxes, and we'll do some experimental work also in the lab to better parameterize these fluxes. And we'll do some settlement experiments also in the next spring, settling for aquaculture based settlement plates, but also settling for restoration using on bottom kind of substrate and preferably shell material. And with this, I'd like to thank you all for listening and say that if you have any questions, please let me know. And also, I would really like to thank those who helped me source the oysters, those who helped me find some space in the lagoon, in the mussel culture, but also helped me a lot with the imports and the logistics. And I'd like to be thankful for those giving me the materials for the oyster cages and the laboratory space, the helpful field work and the maintaining of field equipment. And of course, also to the European Union for funding my Marie Curie project and allowing me to do this work.
Thank you very much, Camilla.
Good morning, everyone, and I'm absolutely delighted to chair this management section with almost 100 participants and at least realize a number of known faces and friends over the years on oyster issues. To those who do not know me, I'm honorary professor for marine nature conservation at the University of Rostock in Germany. But in 2017, together with Bernadette Pogoda, I initiated the first conference on oysters, the first NORA conference, where actually NORA sprang alive as a result of this meeting. So I'm really delighted to have this fourth event now and chair this section. I consider myself personally not as a strict oyster scientist, though I'm a marine biologist, but I'm rather a marine nature conservation manager. For our first presenter, I have some notes, but I know Tom for a while. However, for those who do not know Tom too much, he is an ecologist working at Bureau Wagenburg on marine issues. And he's one of the founding fathers of the native oyster restoration in Netherlands for many years already. And of course, he is interested in facilitation among bivalve species and ecosystem services and reconstruction of those habitats. Tom, you have the floor.
Thanks, Henning. Yes, I'm delighted to see many familiar faces and also many new faces in this meeting. So I wanted to continue on biosecurity, which was taken up by Edo, and also to extend it to aquaculture restoration projects. I see there are shared risks, and I see that as a problem. And also, I think there is a solution which is already presented this morning. Next slide, please. And please note that the background of this slide is the natural settlement of native oysters in the southern North Sea in the Four Delta. And I just want to briefly explain that these risks, these shared biosecurity risks, are insufficiently regulated in EU legislation. And we will discuss how we can reduce them. Of course, as Edo mentioned, we have the NORA's biosecurity guidelines, which is, among others, about diseases, but also invasive non-native species. I will compare the main EU legislation and give some recommendations. Next slide, please. Of course, everyone knows the NORA guidelines. There's a key summary. And I just want to take out one important message that's highlighted in yellow. Always undertake real-time assessments of the sites and oysters or culture material rather than assuming that existing tests and survey results, for example, biocertification, reflect actual status. I will come back to that later. Next slide, please. So the overview already shown by several others. We have native oyster pilots in the southern North Sea, Delta and Wadden Sea. It started in 2015 in the Four Delta, where we had natural settlement, but recently Hein Sassen and I discovered, with the help of some divers, a huge population in the port of Rotterdam, which is started only 2012, and there are more than 100,000 native oysters, all infected with Bonamia and still thriving. And we also discovered a new population in the Wadden Sea. And from recent information, we gathered that this probably originated from native oysters collected by fishermen in the Cleaver Bank and were transported to Texel, and probably are the origin of the Wadden Sea population. So that if that would be so, then the North Sea population is not extinct, but actually surviving in the Wadden Sea. And of course, in the oyster pilots, there are oyster imports from Norway, as Pauline Kamermans told us this morning, and also from Ireland to the Borsele area. Next slide, please. So, as Ada also mentioned, the Bonamia status in the red area in the delta, including the Voordelta, Port of Rotterdam, and Reveling, are Bonamia positive. Which is also an area, the only area in the Netherlands with aquaculture, oyster aquaculture in the wild, while Bonamia negative are all the offshore areas and the Wadden Sea. Next slide, please. But there's also a difference in the invasive alien species. For all the oyster pilots in offshore areas, our company developed developed the license procedure, in which I come back later. And one of the most important risks we had to tackle is the importation of invasive alien species. In all the offshore areas, we have now low impact species like Pacific oyster and the Yassa species, also in the Voordelta, but in the Delta area, we have high impact species like oyster drills. Fortunately, they're still there, limited to only the Delta area. They're not in the Voordelta, not in the Port of Rotterdam. And this is linked, of course, to the presence of oyster culture in the Delta area. Next slide, please. So what about legislation? I think there are three main, with respect to biosecurity, both invasive alien species and diseases. We have three important legislations. That's the invasive aliens regulation of the EU, which is 66 species listed. We have the Habitat Directive, which is linked to the Natura 2000 areas. So for all these oyster pilots on the North Sea, we had to make an appropriate assessment showing that there is no risk of importing invasive alien species. And it has to do with the conservation goals. And it only matters to species in the wild, while the EIS regulation also includes species in captivity in trade. The animal health law, however, only has some listed diseases. It only refers to animals in captivity and compartments in the wild. And so in my opinion, there is no spatial overlap between the Habitat Directive and the animal health law actually in the North Sea, where there is no aquaculture. Next slide, please. So there's also a difference in EU legislation aims. So as I pointed out, the EIS regulation is about biodiversity, as the Habitat Directive, while the animal health law is mainly directed to, aimed at conservation of economical value. Next slide, please. So, but there are common biosecurity risks between restoration and aquaculture. Diseases threaten not only economical value, but also biodiversity, like, for example, crayfish plague and the episodic shell disease in American lobsters, for example. But also invasive alien species threaten biodiversity, like the oyster drills. And the Bonamia parasite is, in fact, an invasive alien species. Next slide, please. And these procedures are complicated. And so as I pointed out, we prepared appropriate assessments for the nature conservation law, for the conservation of the Natura 2000 areas in the offshore North Sea and nearshore of sea. We included for that a cleaning protocol. And so that would be also a good point to include NORA guidelines in the appropriate assessment of these conservation conservation licenses. The animal health law is applied to import license from Bonamia-free areas, which have to be certified. But as I pointed out, does it apply? Because the North Sea is without any aquaculture, and an oyster pilot is not an aquacultural site. And also, is it sufficient? Next slide, please. Because as I started this meeting, this presentation, and also which has been tackled by Sachlikidis' presentation this morning, you always have to undertake real-time assessment of the sites and the oysters, and it is, I think, it's excellent that there's now a new method developed to test oysters alive. Next slide, please.
Tom, you have one more.
That's thank you, and that's the solution. I think testing. So that's the recommendation. You can use also the native oysters in the oyster pilot as sentinel to monitor the Bonamia stage, which is already going on. You can include the NORA protocols in the guideline for the appropriate assessment, and you can develop these non-invasive testing methods, which has been shown by Sachlikidis this morning. Next slide, please. And we have to do more about prevention of invasive alien species risk in aquaculture, I think, because it's currently not included in the animal health law and in the IES regulation. So these species like oyster drill, I think maybe you should also include them in the animal health law and develop specific risk assessment. Last slide, please. So the recommendation is really to integrate the EU legislation. So you have to define species categories which include both invasive alien species and diseases, include the NORA protocols in the appropriate assessments, and implement this new integrated EU legislation. Thank you for your attention.
Excellent. Thank you very much, Tom. I just rush over to our next speaker. It will be Remment Hofstede. Remment is a marine ecologist working as a marine contractor for Van Oord, and he is currently executing a PhD at Delft University. He works on infrastructure in offshore wind farms to support restoration of European flat oyster reefs. And this presentation will be the effect of human interventions at different scales within offshore wind farms. Remment, the floor is yours. Are you there? Yeah.
I'm here, yeah. And I hope you can see my screen.
Yes, we can.
Okay, good. Well, thank you. Yeah, I'm lucky to get the chance to combine my work for Van Oord with a PhD position at the Delft University of Technology. And this PhD is part of the Dutch NWO-funded research project North Sea Revives, which is short for Reef Vitalization for Ecosystem Services. Today I will present a method that we have developed on how we can predict the potential effect of interventions we can take to promote the development of flat oyster reefs in offshore wind farms. And I will illustrate this method with examples of interventions. Yeah, so the method is explained by a framework which basically comes down to this. So we have a situation, in our case, that is wind farms providing a relatively undisturbed environment for marine life, and at least after their construction. And we have an objective, which is the oyster restoration in the North Sea. And in order to reach that objective, we have to take actions. And so how can we do that? Well, the first thing we need to do is to assess that situation, and based upon the historical and the current situation, we can answer whether we can answer the question whether the area is suitable for the desired objective. And second, what interventions can we take to reach that objective? Well, in this study, we have classified 4 scales of interventions, which are the mega scale, the southern North Sea as a whole, macro scale, and an offshore wind farm, a meso scale, the scour protection, which is the rock material placed at the foundation of each turbine to prevent the seabed from erosion, and the micro scale, which is the type of materials that we use in our constructions and the texture these materials have. And then finally, we have to quantify what the effect of each intervention might be. And therefore, we make calculations using the best available knowledge that we have to estimate what the intervention might lead to. And that way, we hope to support decisions that need to be made for the implementation of certain interventions. For example, the selection of certain measures, how many of them, and when and where to apply these measures. Then a brief intermezzo. Many of the interventions, or some of the interventions used in our calculations, they are results from studies we've performed under the research project Borselen 5. I presented the setup of this research project at the NORA conference in Edinburgh 2 years ago, and 2 years later, most of the research is finished. And what we basically have gained from this project is a lot of knowledge on the supply of oysters, where to get them from, how to get them in the wind farm, and on methods and techniques to develop oyster reefs in these offshore wind farms. So the results gained are applied in our framework, but also results from other studies around in the world. So back to that framework. The first thing to do is then to assess that situation. So we all know that oysters were very abundant in the southern North Sea in the past. So the area has the potential to redevelop oyster reefs. And the current rollout of offshore wind farm areas provide also undisturbed areas in which the redevelopment can take place. And in these wind farms, we have distinguished 3 different habitat types. One is the scour protection. It's a rocky material. Next, the other is the soft sediment in between the wind turbines. And the third is potentially layers of shells that need to be placed at the seabed. And we studied the status, the current situation we defined as the end of the year 2020. And at that time, we had 62 offshore wind farms in the southern North Sea, with an estimated total of approximately 3 square kilometers of scour protection and about 3,000 square kilometers of soft sediment. And to put this in perspective, this is still far less than the estimated 21,000 square kilometers once covered with oysters 150 years ago. Well, and then each of these habitats, they can host oysters in different densities. From literature, we have found corresponding densities, a medium density for the scour protection, the rocky material, a small density for the soft sediment, and an extra large density for the layers of shell materials. And in our approach, the area of habitat, of the 3 habitats, and the corresponding densities, they are used to calculate the potential amount of oysters in the offshore wind farms. Well, now that we know the surface of available habitat in the offshore wind farms, we check which part is suitable for oyster reefs. And first we look at the scour protection. Scour protection generally consists of a filter layer of small-sized rocks, topped with an armor layer of larger rock. And the armor layer is stable substrate, the filter layer is not. And from a scour protection monitoring survey that we executed last year, we know that also the species richness at these filter layer is much less than at the soft sediment and at the armor layer. And therefore, we assume also that it's unsuitable for oyster presence. and meaning that the total available of oyster protection habitat for oyster decreases if we take that out of our total habitat available as scar protection. Well, and next thing that we do is we look at the environmental conditions in offshore wind farms, and we can rule out also wind farms that are thought to be placed in areas that have unsuitable conditions, such as discussed yesterday on several talks. For example, by looking at the bed shear stress, and if we do that, we end up again with less habitat available for oysters in these wind farms. So, but now that we have established a certain amount of suitable habitat, we don't have the oysters yet. And as you all know, the offshore wind farms are generally remotely located from existing oyster beds. And due to the limited dispersion possibilities of oyster larvae, these wind farms cannot be reached in a natural way. And to overcome this connectivity issue, we'll need to bring in broodstock to initiate the reef development in the wind farms. And if we assume that we can occupy all available habitat with oysters, we end up with a certain amount of oysters, and of course density dependent, depending on the habitat. And well, this is then our first intervention at the mega scale of the Southern North Sea, so bringing in broodstock. A next example of an intervention is at the macro scale, meaning the surface area of the entire wind farm, especially the sandy seabed. And if we can improve the seabed for oyster reef development, we will get higher densities and more oysters. And a very good improvement, you all know, would be the deployment of layers of shell material. And if we cover a part of the seabed with that shell material, the total amount of oysters in the wind farms, they will increase significantly. Don't mind too much about the numbers because these are all estimates, but just look at the colors. Green is positive, red is decrease, and yellow is orange is no change. Well, and if you do that with the shell material, you also should take into account, of course, that it's not always available in the large amounts that you need to come to significant amount of additional oysters. Well, at the mesoscale, we can also take an intervention. The mesoscale we define as the scar protection. Can take many interventions actually, but this is an example. And the density of oysters at scar protection is assumed to be higher than at the surrounding seabed. So if you increase the available scar protection, the armor layer of it, in the wind farms, you would likely also, that would likely lead to a higher amount of oysters. Another intervention aimed at the scar protection, but then at the micro scale, is to install a top layer of calciferous rock, such as marble. Studies indicate that also under the Barcelona 5 project, that marble could lead to a higher ratio of larvae settlement, namely factor 1.3. And we assume here that a higher settlement rates of larvae will also result in higher densities in oysters at the scar protection. And it's also important to mention, I think, from a construction perspective, that such an intervention of optimizing rock material can be easily implemented in practice without additional cost for purchase or installation if you use the same type of equipment as for installation of conventional scar protection. So it's therefore very much worth investigating the actual effect of such an intervention. Well, to conclude, we are aware that with this study we make many assumptions, and by no means we claim to predict the precise numbers.
1 minute, please.
Well, I'll manage within that minute, thanks. We don't claim to predict the precise numbers of the percentual oyster quantities in these offshore wind farms, and well, let that be said. But however, we do believe that we provide a tool that allows comparing the effect of different interventions using the best available data, and also to estimate how much of a certain intervention is needed to reach a desired goal. Well, of course, these calculations, they can be fine-tuned over time by using the most recent knowledge or by using location-specific information. And also we can add other interventions that I did not include in here, but or interventions that are even not yet developed. So we hope that our approach can support decision makers and wind farm developers in the selection of measures to reinstate oyster reefs in the North Sea. Well, yeah, this concludes my presentation. If you like to have further information, please contact me or have a look at videos of which the links are provided on the screen. Thank you very much for your attention.
Thank you, Remment. And we move straight away on to our next speaker. It is David Donnan from Nature Scotland, and he works in the Sustainable Coasts and Seas team of that agency. He is a governmental advisor to the Scottish government and works in focus on biodiversity and marine fisheries interactions and on non-native species. He has been involved in native oyster conservation in Scotland and also with us at NORA for many years, in Scotland for over 20 years. So David, we are delighted to listen to you. The floor is yours.
Good morning, everybody. Thank you, Henning. Nature Scot is the statutory nature agency in Scotland, and we've got about 30 years' experience of advising the Scottish Government. Our work at the moment is about enhancing the natural environment and inspiring the people of Scotland to care more about it. We're working towards the green recovery from COVID-19 and looking for a nature-rich future in Scotland and an effective response to the climate emergency. Nature Scot plays a largely advisory role, although we do have responsibilities in species licensing and including translocation for conservation purposes. That's obviously relevant for our oyster work. Our colleagues in Marine Scotland are the Scottish Marine Management Authority and regulator, and in relation to consenting marine activities and for fish and shellfish health. And collectively, we've had an engagement with all of the projects running in Scotland at the moment in some form, but that varies from simple contact through providing advice and to actually engagement to the extent of providing funding and involvement in the projects. So the conservation of the wild native oyster population is the sort of central theme to my presentation today. And we've been working since the 1990s on the conservation of oysters in a number of ways. First and foremost, just about building the evidence base for the distribution and abundance of oyster in Scotland. Working with partners to control illegal harvesting, particularly about 10 years ago, and listing the native oyster as one of our priority marine features and making sure it's been included in the Scottish MPA network. And there's also been a body of work in looking at the feasibility of oyster restoration. Right now in Scotland, there's a variety of oyster restoration projects underway, and many of them are represented in the audience today. So just thinking about the characteristics of the project so far, there's a number of factors at play. One is diversity in terms of their age. You know, we've got discussions about restoration going back at least to 2007. And for the Dornoch Deep project, that's really 2014, I think, you know, that's been running for. And we've also got projects that are just at the proposal stage or very recently started. There's also a lot of variety in terms of the scale and the resource being applied to the projects. And these range from complex partnerships with funding in place, industry and academic involvement, right through to small local community initiatives with little funding, but lots of enthusiasm. And then the complexity in terms of the approaches and methodologies being used are suggested, you know, ranging from deployment of broodstock cages, culch and oysters onto the seabed. And as a result of that, the projects trigger a number of different pathways depending on the methods and their size and their location, and you know, as Tom was reflecting on in his presentation earlier. At the moment, there's no one-stop shop or single consenting pathway that's in existence. And certainly in the last year to 18 months, there's been an awful lot happening in terms of these projects activities. And this particular NORA conference has been good timing to take the opportunity to pause and reflect on this and think about where we're going and how best to get there. So my purpose today is not about sort of saying, sorry, how this should be, but you know, to offer our thoughts on the fundamentals about what we need to get right, and so that we as a community of folk interested in oyster restoration and conservation can have the necessary dialogue and effective working to achieve our goals. So with that in mind, the first defining factor for us, you know, is the presence of our wild oyster population and the need to ensure that what we're doing in restoration and enhancement work doesn't inadvertently become a risk to it. The current distribution of oysters in Scotland is the west coast and the islands, the Western Isles and Shetland and Orkney. And that has implications in terms of a spatial dichotomy and what's required on our two coasts. So on the east coast, what we're talking about really is restoration of the former range of the species. Whereas on the west coast, the task is about enhancing the wild population. But it's true to say that, you know, the understanding of that wild population on the west coast is really far from complete in understanding both the distribution and abundance. And addressing these uncertainties is important for a number of reasons. Firstly, in thinking about informing the siting of enhancement work, both locally and nationally, relative to the wild oyster population, to avoid the potential impacts on it, whether that's through genetic disease or non-native species routes. Currently, the projects are located largely according to where there's interest and enthusiasm, and rather being based on a more strategic consideration of siting. So with respect to that, taking that forward and for building the evidence base, I think there's definitely a role for both the authorities like ourselves and for the projects. And we're seeing that through a range of both formal and citizen science survey efforts to improve this information. But there's also a link to another one of the fundamentals, and that's the requirement for securing a genetically diverse broodstock to supply the restoration and enhancement projects, as we've heard mentioned already this morning. And of course, the wild oyster populations are of tremendous value for that. So at this point, it's worth mentioning some current action which is underway. This is the Oyster Ark project, which is funded by the Scottish Biodiversity Challenge Fund, which NatureScot administers. And this is lasting the current financial year until the end of 2022. The lead partner is Heriot-Watt University, Bill Sanderson, and it's really about addressing two fundamental objectives. First of all is further review and new quantitative survey of oyster beds, to help build that evidence base. And it's looking at the development of broodstock and towards setting standards for the stock that can be used and supplied into restoration. So I'm just noting this here as a work in progress, and something that Bill, I'm sure, will follow up with results in due course.
You got that? 1 minute.
Okay. So I'm just going to spin very quickly through a couple of the other fundamental things. One is that we need to embed, you know, a adaptive management approach in what we're doing. With this diversity of projects underway and the range of uncertainties, we need to keep this cycle of monitoring, assessing our actions, and acting in accordance. And from our perspective, the monitoring really needs to be able to detect if the wild populations are being improved by the enhancement work, and also to detect that disease and INS issues at the earliest point. The final fundamental is really about timescale, and this relates a lot to funding. At the moment, a lot of the funding is built around short-term funding opportunities, and that's inevitable. That's not a criticism of the of the project. And seeking to build a longer-term stability into funding is a key thing. And there's steps towards that through next year, the development of the Scottish Marine Environment Enhancement Fund, and we're already plugging into other funding opportunities in Scotland as well. And also taking advantage of the changing management and legislation landscape to embed this longer term perspective in what we're doing, and not least the Scottish Biodiversity Strategy next year following the COP15 conference on biodiversity. So I'd just like to finish with what I think success is going to look like for us. Firstly, it's about the native oyster being restored to its historic range in Scotland, and this being part of our contribution to the recovery of the species across the North Sea basin, and that we've built a robust wild oyster population across the West Coast and islands without having negative impacts on the wild population that's existing there already. And that in doing so, we're meeting our targets for halting and reversing biodiversity decline and building resilience, especially for climate change. So thank you very much for your attention.
Excellent. Thank you very much, David, for this brilliant insight in Scottish nature conservation-driven oyster restoration intentions and activities. Our next speaker will be Rob Whitley from Natural England, and he is the National Specialist in Shellfish and Aquaculture. So Robert, if you are ready to go, the floor is yours.
Henning. Yep. Thanks very much. Can you see my screen okay?
All fine.
Yeah, great, thank you. Yeah, thanks for the introduction. Yeah, Rob Whiteley, Senior Specialist in Marine Fisheries team, Natural England. Just wanted to give some feedback and perspective from our experiences in native oyster restorations as England's statutory nature conservation body. Look at a couple of case studies and considerations around legislation. Take-home point is it's been a big learning exercise for sure. We've been kind of finding out as we go. I'm going to spin through some bits here. These are just some of the key take-home points, but obviously can provide more information around any of them. These are some of the kind of main messages. So the first one, it's the what's known as Blackwater Estuary. It's the site of the NORA restoration project as well, which you'll have heard more about. But the main thing here, it's a multi-designated site, but it includes MCZ, a marine conservation zone for native oysters as a species and as a bed, both with recover objective. The bed designation is 5 per metre squared, which was taken from the OSPAR definition. That was the default one to kind of use. But we found over time this was proven unsuitable for the site as a restore target, primarily because of the presence of Bonamia and obviously the increasing densities and the high risk of that. And CFAS, the government kind of scientific advisory body, were recommending, I think it was 1.26 per square meters for the site. And there's naturally occurring lower densities in this site. So the recovery target was an objective, a trajectory to move towards. And given this was over time, the experiences and feedback on this, we found it was making our advice to the regulatory body, the IFCA, difficult around this. It was proving hard to kind of justify and determine. So I went back about 3 years ago now, contacted OSPAR, asking about the bed definition, and they confirmed the 5 per metre square didn't account for Bonamia conditions. There's no variation or variability in that at all. It was just a kind of definition picked at the time. And then more recently, we commissioned a review of defining the beds or habitats in the site, primarily undertaken. It's Tom Cameron, who's presenting later. What this did, it confirmed lower densities in the site, but that's on average. So this was long tows. This is average densities with patchiness established. It recommended a 1 per meter squared minimum. And what we're doing now, based on this review and again previous experience and evidence, we're looking to change the conservation advice on this site for this definition only, and that will be published in March. Again, we just feel it's a more appropriate target, and it reflects what the site seems to kind of suggest is sensible there. Couple of caveats: it's based on current understanding. This can change, our evidence and picture can change, so could our advice. And it's based on assumptions on dredge efficiency as well, which I think Tom's talking about later. Okay, this is the Solent. Luke gave a really good review of this yesterday, of the project there. This is the main site at the top left, bottom right. There's also a small MCZ for native oysters as a species. So again, it's a blue marine foundation, and again, like Blackwater, it's a multi-designated site. So there's a lot of conservation objectives to consider here. When the restoration was proposed for the site, it's how do we take what's being proposed and put it in a shape where it can fit in between and within all these designations. It's a tricky balance and something we had to put a lot of consideration into, especially the area team. In short, really, we advise it could only occur on the areas of subtidal mixed sediments within the sites. And the Blue Marine Foundation surveyed this as well. They did some side scan and grad samples just to determine where the subtidal mixed sediment features end and the other ones begin. And also a key thing here is the historical presence of native oysters on the site. That was a key thing, because there is a specific objective for native oysters on subtidal mixed sediments feature. It's not designated for this particular site, but we feel it's one that would have been there historically, so it wasn't uncharacteristic of the site, and it's something that wasn't kind of countering any of the objectives. It also helped to determine the spatial extent of the restoration. So it was 0.15% of the total SAC and just over 1% of the subtidal mixed sediment features within that SAC. So again, it just when you're looking at the evidence and making the determinations on the advice around it, it just helps to kind of quantify and give it a rationale. Another factor was it's a capitular dominated site. So all else being equal, it just gave us less concern about the habitat in question, because it wasn't kind of in pristine state, and just less concern. Another requirement, I think it was the same for NORA 2, was that the culture matched the sediment found on the site as much as possible. So it was kind of a characteristic one and wasn't creating effectively a different habitat. So this has taken us more widely into some kind of legal considerations, with the potential to help restoration projects, any future ones, get off to the best start we could. So again, looking at where you've got subtidal mixed sediments, where you've got the historical presence of native oysters, it's the best place to start. It's not an open door. It just means within MPAs, it's going to be the easiest ones to kind of defend and justify kind of going in, and it just makes it kind of an easier legislative passage. Even easier would be to outside of.
Rob, 1 more minute.
1 minute, okay, thank you. Even easier would be to be outside of MPAs, but we're talking inside here. But obviously, there's due process, so we've got statutory advice we provide, best available evidence. We need to balance it again with other site objectives. We've got the potential to alter conservation objectives within a site, so if one was less favourable, we can include a native oyster definition. And these are different considerations to dispersal-based introductions as well, which is a different thing. So just to boil that down, this is England's full suite of MPAs. If you can see my cursor, this is obviously Wales on the left there. It's not an absence of designations. These are the sites with subtidal mixed sediment. This is a subtidal mixed sediment mapped out. These are the SACs. These are the MCZs, and these are the SPAs. So again, any area outside of those that's an MPA, it's going to be just a bit harder for us to defend it going in there. Looked at favourable conservation status too, going back a couple of years. now, but this is it's within the Habitats Directive. It's a key set of principles around wider goals and objectives for species and habitats. We've mainly adapted it to terrestrial features, but we've tried it for native oysters and spiny lobsters as well. So it's been difficult because you're trying to find the ideal end goal, but you also in your head you're looking at the evidence that's stopping that, which is particularly the case for native oysters. So the key factors, it's no loss of self-sustaining populations where they're present, self-sustaining subpopulations restored, again looking where the historical presence is, and then determining density targets like we just discussed. That's been informed by that sort of review. And it's confounded by data gaps on the status of the species and the quality and extent of supporting habitats, which on the mapping we have, it's not clear, especially where they're historically present. More work there. And then last slide, really. So some of the considerations we've learned, I mean, partnership working is key, especially at the local level and especially at early engagement. So for any restoration projects, it must be across the board, really. Timings, you know, there's due process involved in all the assessments, and there's timings involved in that, which may not necessarily align with things like seed availability and whatever else. So speak as early as possible. We can start to see, as I put here, the snakes and the ladders, the things that are going to kind of hinder and help any restoration potential. Data gaps are key. Again, you know, it's transparent evidence-based decisions. So we need to find out where the data is missing and look to generate that. And I do think, lastly, from our perspective, I do think we've been supportive as we can within our remit. So normally for commercial operations, standard stuff, we charge for pre-application advice. So before it's formally submitted, we'd be charging for any advice we provide. And we've offered a lot of early engagement in this because we see it's no commercial enterprise. And more widely, lastly, we are considering what we want from our seas coast.
Time, please. Rob, remind the time, please.
Yeah, sorry. This is the last point, sorry. So what we want from our seas and coast, so it's looking at wider targets around links to blue carbon, etc. Many thanks.
Okay, thank you very much to Rob as well. So thanks for this wonderful session, and I look forward to meeting you again in 1 hour.
Hello, everybody. Welcome to the afternoon session focused on monitoring. Philine, just going from my notes, you're delivering the restoration handbook first.
So my name is Philine Ermgassen. For those of you who don't yet know me, I am part of the NORA Secretariat. And if you're new to this meeting, thank you for joining us. I'm delighted to be presenting on behalf of the NORA Monitoring Working Group and the Native Oyster Network UK and Ireland, this product that's been a long time in the coming. It's been over a year and a bit to work pulling this handbook together. And today we're very pleased to be launching the European Native Oyster Habitat Restoration Monitoring Handbook. I also want to thank all of our funders and supporters for making this work possible and in increasing its reach as well. And they're all listed at the bottom. But as I mentioned, this has been a phenomenally large and long effort. And I should first of all recognize the great contribution of all of the editors in pulling together so many pages of exact science in an accessible format. And also to all of those experts who've contributed text, review, and their knowledge to these monitoring guidelines. The rationale behind wanting to pull together some monitoring guidelines for native oyster habitat restoration were that as we've seen in recent talks, there's really been a phenomenal growth in native oyster restoration efforts across Europe. In 2015, there were just 5 projects. And as of now, I'm aware of 33 projects that have made themselves known to the NORA network across Europe. And with this growth is a huge amount of knowledge being gathered and gained and a huge number of lessons being learned. And it's really important that we try and learn these lessons together so that we're not wasting resources unnecessarily. Monitoring brings with it many benefits. It's challenging, it's costly, it's time consuming. No one is denying there are drawbacks to it, but there are huge benefits that can be realized in both the short term and the long term. In the short term, it provides us with information about project progress And in the longer term, it provides us with an evidence base that we can use when talking to stakeholders and engaging with people outside of native oyster restoration to explain what it is we're doing, why we're doing it, and how it progresses. And really importantly, also for policymakers, understanding that evidence base so that it can be used in formulating policies moving forward. So how we went around doing this, we met in person in December 29, pre-masking and travel restrictions. And I think of it very, very fondly now. We were invited to Portsmouth by Joe and Preston of the University of Portsmouth and the Native Oyster Network UK and Ireland. The monitoring working group was invited to participate with that network in contributing ideas, thoughts, text on what the monitoring metrics should be, what it is that we should be looking for in our native oyster restoration when we want to think about reporting. The next stage was then to draft text, and that text then went to open consultation in May of this year. And through this consultation process, we gained more guidance, more authors, more knowledge, and we can again only thank all of those who downloaded the document. It's hefty, it's not a light read, or at least it wasn't. It's very smooth now, but at the time it was glitchy, and it's been really massively improved by those people's efforts. And so thank you to those who did engage with this process. And today we can show you the final shiny version with thanks to Celine Gamble and Azra Glover for seeing it through to these final stages of design. It's been kind of a large task for them over the last weeks. And I hope that you will go to the project zone in Gathertown after this meeting and download your own copy from the project store. It's also going to be available, it is already available on the NORA websites and on the Native Oyster websites. So there's no reason not to go down and grab a copy for yourself. What I'm going to do in this presentation is just provide you with a brief overview of the 5 chapters that we have included. So chapter 1 is a background text. It's text that's there to provide those new to monitoring or new to native oyster restoration, the science behind it, with essentially a leveling text that provides with basic information for understanding what we mean when we're talking about monitoring. So it gives information about random sampling and quadrats and transects, but importantly, it also provides us with some background information about what we mean when we're talking about native oyster habitats and what they might look like. That's been a really long and involved conversation, and we're very pleased to have been able to lean on this work from Stéphane Bouvreau in France, because in France, we really do see the whole range of habitats that native oysters provide, which is not the case throughout its range currently today. Chapter 2 picks up on the universal monitoring metrics. And by universal, it means that the experts who drafted this text believe that these are metrics that all projects should be monitoring and recording. They are deliberately designed so that they should be relatively light touch in terms of effort required, but they yield the best information about being able to tell your own project supporters, but also the world as a whole, about progress that is being made in native oyster restoration. Those universal metrics include being able to describe what the area of habitat that's been restored is. As we know, native oyster habitat isn't just live oysters, it's also the shell cover that they provide and contribute in the longer term. So metrics around shell cover, but obviously also living oyster densities are included. And for thinking about progress and how that habitat is developing, also understanding your oyster size frequency is listed. The last 2 metrics relate to environmental variables that, again, across its range can help explain why you're seeing the kind of habitat that you're seeing. And those are including temperature and salinity. And what was really interesting in these discussions is that we call these universal monitoring metrics, but it was kind of affirming through these discussions to realize that these guidebooks were really necessary. There was some differential use of terminology, for example, that we're hoping to be able to support the community in clearer communication moving forward around things like shell height, for example, and around footprint and area of habitats. The third chapter, relates to the supplementary monitoring metrics. And these are metrics that are a little bit more involved, but they provide a huge amount of information, again, about the progress of oyster restoration. They include things like growth rates, survival rates, larval abundance and fecundity, and disease prevalence. And while it seems like an awful lot of metrics, they're summarized in this infographic, and you can see that a lot of the metrics can actually be yielded from a relatively small sample of oysters, and otherwise there's recommendations as to methods in the field or in the lab that will help yield that information. It's not just slick infographics, although we are very pleased with how they've turned out following the many iterations and development in terms of trying to make them as clear and as accessible as possible for all readers, and not just those who are already familiar with monitoring. But there's also really detailed methodologies and useful photographic notes illustrating how these things might happen in the real world, how you might go out and look at histology or gonad ripeness or sediment traps. So we really hope that when you visit these, you find that they are useful and provide you with the guidance that you might be seeking within your projects or within projects that you know. And the fourth chapter relates to restoration goal-based metrics, and they seem very heavy in the list that's here, but actually they can be summarized as being metrics that relate to understanding what the benefits are of native oyster restoration. And many of these are still fields for which we have very few data. So we're hoping that, again, that these guidelines will provide a kind of starting point to make sure we're all speaking the same language when we're collecting this information. There's information on larval overspill. So thinking about the broader impact of your oyster restoration site on oyster potential fisheries nearby, for example. There's also information on the biodiversity and non-oyster fisheries and invertebrates and how you might quantify those. Benefits to other coastal habitats like seagrasses and salt marshes, the potential benefits that you have there. Benefits to water quality, socioeconomic benefits, and finally carbon in terms of both carbon stock and a carbon sink. So methodologies for all of these are outlined in the guidelines. And they're outlined in a way that's intended to be very accessible so that you can go out and really plan your restoration project, or your monitoring rather of your restoration project around gathering this information. And again, the more sites we have this information for, and the more they talk to each other in a language that is common, the sooner we can build up this evidence base and move forward with the science as quickly as is reasonable. And the final chapter is on evaluation and reporting. This is a very brief chapter, but it's intended for those who are, again, perhaps less familiar with restoration monitoring and with science in general. So it's thinking about not just collecting the data, but what you do with that, how you store it, and how you analyze it moving forward and report it ultimately to your stakeholders or user groups or the wider community as you see it. Because it is quite a hefty document, and it takes, I mean, sorry, in a positive way. Sorry, I know that sounds negative when I say hefty, but it's really detailed. But to provide it in the most accessible format possible, we've also put in a overview summary table at the end, so that if you're interested in a particular metric, you can at one glance understand what the content is in terms of what methods are being recommended, what alternative methods are being recommended if those initial primary methods don't suit your needs or don't suit your site. What units they should be reported in. Again, this is important. How often you should be thinking about going out to sample those. And finally, also performance criteria is assigned to each metric, which is essentially saying, should your value be going up or down or staying the same in order to show that you are progressing or achieving what you would like to within your project. This guideline, this guide handbook forms part of our broader restoration guide family that's been co-produced between the networks in terms of the restoration handbook for the native oyster network, where the NORA network was involved in the biosecurity chapter specifically, the biosecurity guidelines, and now the habitat restoration and monitoring guidelines. And I hope you'll tune in later to our site selection working group roundup as well, because Anna Hughes will also be presenting some further exciting advancements from the community in that project. Again, a reminder that these are all now available for download on our website, so please do go and take a look. And I just want to finish by thanking again the phenomenal effort of our editors and our authors in putting this together. And thank you all for listening. And thank you, David, for reminding me that I was speaking now.
So I'll just start the session very quickly. I mean, basically, there couldn't be a better introduction to this session than the launch of the monitoring handbook. So congratulations to everybody involved in getting that published. It's going to be of great value to the full range of people involved in oyster restoration, from the professionals through to citizen scientists. Today's session is a sort of mixture of feedback on results and on methodologies. And, again, sitting at our desks, it's an opportunity for some virtual traveling. We're going to be going to Ireland, to the southern North Sea and around the UK. So I'll just introduce, to start with, Oliver Tully. Oliver coordinates oyster stock assessment and oyster restoration projects in Ireland in collaboration with a variety of industry and community partners. Over to you, Oliver.
Thanks, David, and thanks for the opportunity to speak to you. And I presume you can all see my screen, so it's just an update, a kind of a whistle-stop tour around the west coast of Ireland to update you on the activities of the various restoration projects that are going on in that area. I've put kind of restoration in brackets here, or in quotes, because really, I suppose we're involved more in enhancement and management, rather than restoration from a blank sheet, as it were. We have active kind of spawning populations of native oyster in all the sites we are working in, and it's not to say that they're in a truly functional state. They're, if you take Stefan Bouvreau's categories, we're probably somewhere between fragile and favorable, and maybe even functional in some cases. Although we reserve that one, I think, because even where we have high fishing or high oyster densities, we also have some fishing activity. So whether we have true we can optimize the ecological function here is still open to debate, really. The projects are very much collaborative across a whole host of people and organizations. The Institute kind of tries to coordinate the scientific aspects of the program, and alluding back to what Philine was saying, to ensure we have at least the basic metrics that we can report on to provide the evidence base behind restoration. And the projects have been going on since maybe 2017, later in some sites, and we're working in 4 sites at the moment. That's Galway Bay, Kilkieran Bay, Clew Bay, and Lough Swilly. And all of the organizations in those areas have some management authority over those areas. Well, not all, but some of them have some devolved management authority for oysters in those areas. And in some cases, that's not the case. And we're also working with our kind of sister agency, BIM, who's responsible for fisheries and aquaculture development in some of these areas. So it's very much a collaborative effort across a whole host of people. Is the slide progressing? Yeah. Okay, so I'm going to show you 2 slides from each site, so you'll know I'm progressing towards my 10-minute time limit. So in Galway Bay, research for restoration or enhancement, our current status and evidence base here is, well, we have spawning stock. We have very high numbers of very juvenile oysters in some areas. We've been, distributing culch. In some cases, we have very weak settlement on culch. We have estuarine low salinity problems in some areas. We're monitoring Bonamia. It is present. We've had no fishing for at least 5 years now, but we still think mortality in this area is high. So if you look at these, the areas here of activity, we're involved in habitat suitability assessment, enhancing settlement through habitat manipulation, getting empirical data on survival and growth of known age oysters, Bonamia monitoring, of course, baseline kind of survey data, and also outreach and outreach activity. So that's the landscape we're looking at in the map on the left, a mix of native oyster production areas and Pacific oyster cultivation. In 2020, we scaled up our kind of culture deployment quite significantly from experimental scale to kind of what you might call, commercial, or, at least a significant scale up from that, and deployed about 500 tons of mixed scallop and crushed whelk shell. And we've been able to monitor that using acoustic methods and providing the, to what extent we've developed relief above the basic seabed level, and to quantify the area and extent of the shell that we've deployed, and then subsequently monitoring of settlement onto that shell, which would give us a population estimate for the settled spat. We have a new kind of very high resolution hydrodynamic model for the inner bay where these oyster sites are, which enables us to model the distribution of salinity and temperature and combine it with experimental data on the response of oysters in this system to variation in temperature salinity. So we have like dose response kind of relationships to combinations of temperature and salinity we can use in the model to define suitable habitat for a native oyster with respect to salinity and temperature. It's kind of all in the numbers then, isn't it? When we look at survival growth and whether that projects into increasing biomass in these systems. So we're looking at known age oysters we've got from onto spatted culch and spatting ponds, deployed them intertidally and subtidally, and we're having monthly estimates of growth and mortality from those sites. In Clew Bay, Midwest Coast, we also have high spawning stock and really kind of high density oysters in some areas, very aggregated distribution, good survival on spatted culch, also Bonamia monitoring. Surveys, of course, to provide the basic evidence of the distribution and extent of oyster beds present at the moment. And again, like in Gaul, we have empirical data on survival and growth of early life history stages or early year classes. And again, outreach and education is important in this area. So in Clew Bay also, we're modifying habitat by removing of silt from shell prior to settlement. There has been, of course, a lot of discussion about whether harrowing is beneficial or the disruption and cleaning of the shell is important prior to settlement. In this case, we're not really disrupting seabed, but simply removing a superficial layer of silt that settles onto shell during winter and spring, and having that removed prior to settlement in these areas. High distribution, very dispersed kind of distribution in this area, and never do an oyster survey in Clew Bay without some local knowledge, because you will get lost. It's very complicated topography, and also complicated to assess the distribution and extent of oyster beds in the area. We have some very high density areas in some places, and we have also fishing in some places as well. We have also areas that are close to fishing, and we're also looking at survival and monitoring of spat. There's a lot of outreach activity there as well, involving students in collaboration with BIM and the local co-op, and that's been really beneficial in getting extra data and extra boots on the ground to do this work. In the north of the country, in Lough Swilly, the big issue in Lough Swilly is the very, very high abundance of Pacific oysters. So they're cohabiting with native oysters. But nevertheless, there's still some good settlement of native oysters in this area. We're performing kind of, the same type of work. We're Bonamia monitoring distribution of biomass of both species from surveys. We really need here a better grasp on fishing activity and to develop what you might call better fisheries management planning and better legislation, which enables us to separate out these two species in legislation. The legislation governing the exploitation of oysters and management is old and predates the introduction of Pacific oyster, in fact, into Ireland. So that's a problem at the moment. We have areas here in Swilly where we are looking at the growth and mortality of native and Pacific oyster where they're cohabiting. We have areas where we have culch deployment, and also areas where we've relayed juvenile native oysters captured in the Pacific oyster fishery, relayed them onto clean sediment. Very interesting outreach and education activity here through Fiona Stapleton at PZ Maps, where she's using. She brings children to GIS school and using basic oyster surveying methods to teach them about GIS and mapping methods in the marine environment. Very useful initiative indeed. I think there's
just a minute, Oliver. Thank you.
Okay, that's fine. In Kilkieran Bay, the big issue in Kilkieran Bay is this complicated network of habitats that we have within the system, and that even doesn't appear to be static. So we have a mix of sensitive habitats, seagrass and marl, and intermixed with oyster. And so we're concentrating efforts here in subtidal sedimentary habitats with respect to oyster enhancement and restoration. Again, enhancing spatfall using cupels and culch in this area strategically in areas which are subtidal sedimentary habitats. But there's a big discussion there about the conservation objectives, and we've heard that earlier today about the consistency of restoration with conservation objectives within SACs. So in summary, it's important to really, given that the effort required to restore is really significant, and it's difficult to reproduce what nature is already doing. So that tells us really to protect what we have, and that's the primary thing we should be concentrated on. There are a whole other range of work we're trying to get involved in site selection and optimizing where to put our restoration efforts, population modeling and carrying capacity models, and some of the work that Brecht was talking about yesterday is really important in that respect, I think. The question really for us now is trying to translate from experimental phase work to scale up. And that decision process involves a lot of questions and a whole other set of targets and set of objectives and a whole different even set of stakeholders perhaps in trying to make that decision, and on what basis you make that decision and when you make that decision. And we haven't made that decision. We haven't really made that decision, but I think we need to grasp that question sooner rather than later, because we can't continue indefinitely to operate at an experimental scale. Thanks, David.
That's great, Oliver. Thanks very much indeed. We'll have to move on quickly to Tom Cameron, who's a senior lecturer in animal ecology at Essex University. He's an interest in how populations and communities respond to environmental change and predation and management. Tom's research group has aquatic ecology focus, and his current projects ranging from coastal fisheries, aquaculture, marine and freshwater habitat restoration and management. Over to you, Tom.
Thank you very much, and thanks for having me this afternoon. So today I'm going to be speaking about a very applied piece of work that emerged from lots of discussions between oystermen, NGOs, researchers, and government involved in the Essex area on projects to do with native oysters. And this is to do with estimating the efficiency of a dredge for estimating the abundance of native oysters on different habitats. So there's lots of different ways to assess the abundance of a shellfish on the seabed, and they'll have their pros and their cons. Some of the most preferred ways, maybe for conservation objectives, are grab sampling, because they have less effect on the seabed. And the benefit is they're very highly efficient. You get every organism that is inside the grab, but the cost is very low coverage. You need lots of those grabs to compensate for that high efficiency but low area, and it needs that actually high density to meet that cost benefit analysis. The last time they were recommended in a report that was dealing with 18 to 30 oysters per meter squared. I'm not used to dealing with those high densities. Another way that's becoming more popular is using video or technology, diving and photographs, and also now sonar and even lasers, laser sonars, to be able to see the shellfish on the seabed. And they're very low impact, but they have relatively unknown efficiency in all conditions. In some scenarios, they can be highly efficient. We saw another contribution from Luke Helmer yesterday from the Solent. This is an image from some work, a lot of work done with videos up in Scandinavia recently, really effective. But this is what my seabed looks like, and it's not, we've tried video and it's not as efficient in our highly sedimented system. Dredging, we're all familiar with. I'm talking here about a ladder dredge where the front of the dredge has a ladder on it, and the oysters either go through that and are not captured, or they're bump off that up into the net above and they're captured. This is a very large surface area, so it has a benefit. It captures even when the oysters are thought to be at low density with relatively low effort, but it does cause damage to the seabed, if you want to call it that. And so therefore it might not meet with the conservation objectives. But it is still widely used for various reasons. But it's used with variable estimates and assumptions of its efficiency, despite people using the same dredge for the same organism. This is largely a look at the UK landscape, but I have looked at some other species fish forward ladder dredges, and you can see that efficiency assumptions vary a great deal. So even within the IFCA organizations, but between different regions, different efficiency estimates are used. Southern IFCA started off reporting using a 5% assumption, later switched to catch per unit effort because they didn't know what assumption to make. Cornwall IFCA were publishing from 2015 to 2018, but not quite reporting what efficiency assumption they used. It seemed to be that we're using 100%. Kent and Essex IFCA, working with Natural England and ourselves, settled on a 20% value after a literature review starting in 2016, accumulating in 2019, with a range of values of 10 to 30%, representing what might be experienced. We see some early work on Ostrea chilensis and of 15 to 18%. In Northern Ireland, can range from 5 to 25. And as you can see, there's just a whole range of different values that are used. And this is important. It does matter because there's huge potential for error if these values are used to extrapolate from catch up to estimated population size. In Essex, we're dealing with an area of 284 square kilometers, so we can't help but extrapolate. So we must get an accurate estimation. We could be dealing with, underestimating or overestimating our oysters by 4 or 5 times the actual value. It matters for these surveys then, and these surveys are what we use, and these data are what we use to make models to predict recovery or to predict quota. So it very much does matter. And it also matters from the interaction with other stakeholders. Oystermen claim often to me that there are more oysters on the ground than I say that there are, and they say that's because of our dredge efficiency assumptions. And so it's been some time since we've had experimental work on this, and this is exactly what oystermen locally in Essex asked for at a workshop in 2019. And working with them at that workshop, we got a flip chart open, and they told us they wanted us to see working on ground types, how efficiency is affected by density, by the distribution of animals, if oysters are clumped or more uniform, how the season in the year might affect it, and how numbers of dredge passes. And so we took that away and we tried to secure some funding. Thank you, Natural England. They helped fund this work, and we went about resolving this work. So we found a site that was a lowest intertidal site in the Essex area. It was a several order, which legally permitted us to do this work, because otherwise we wouldn't have been able to do it. And it was a site with no or few native oysters on them already. And it was safe to access at low tide on foot. Various ground types, but I'm going to class them as very, very hard, such as this one here in the foreground, to softer ground that had layers of soft mud on them. We were able to mark out the beds, if you like, that were going to be dredged, and all the oysters in the study were catchable because they'd previously been caught elsewhere in the river by these same ladder dredges. We knew they were catchable oysters, and we could control the density of the oysters on the beds because they were hand placed. It was a fully controlled experiment. So going into results, first of all, did the method itself actually work? Well, yes, I think it did. I hope you can see from these images here, you can see very clearly after returning to the low tide site, the site at low tide after it's been dredged, you can see the dredge line occurring here, just to the right of the end marker post here. There's another post here. And you can see the dredge marked where the dredge had passed over the area where the native oysters were present on the bed. So the intertidal access permitted the checks on the dredge activity, and there were only 3 zero catches in the whole study, and they appear to be real zeros where the dredge passed over the oysters, but it still didn't catch them. What about overall dredge efficiency? Well, our overall mean result is 10.7%. It didn't matter whether we had whether this was the first pass or the second pass over the site, and it didn't significantly matter between seasons. What about the ground distribution? Thanks, David. The distribution of the oysters on the ground, or the hardness of the ground? Well, there was an interaction here. So if the oysters were evenly distributed, the efficiency increased as you moved to softer ground, but this wasn't the case when the oysters were clumped distribution. And what about density? Well, there was an effect of density. So as the density of the beds increased from 1 oyster per meter squared to 2.2 meters squared, the efficiency declined. So this means that the dredging efficiency is density dependent, which is really important to note. So catch appears to be density dependent. This means that catch per unit effort methods for using oyster ladder dredges are probably not appropriate. If you find density dependence in catch, it means that these methods could be dangerous for setting quotas, for example. The dredge efficiency was also affected by the distribution of the oysters and the ground type. And we could witness this at low tide, that on soft ground, as if the oysters were uniform, the dredge approached them and it would scoop them up because the dredge would go through the mud. But if they were clumped, it was like taking something through the soft ground, and when they hit the hard clump of oysters, the dredge would bump over them and push them further into the sediment, reducing their ability to be caught. So there was an interaction there. It wasn't affected by pass or season, but it could have been our methods. We only had 2 passes in our dredge in the study, and everyone knows that when oystermen are actually fishing, you would have multiple passes. So this is probably appropriate for a survey dredge result, but maybe not for fishing. And the distribution was really important. We need to know how our oysters are distributed on the seabed. They're almost certainly not uniform. And so understanding what that might be will be important in the future. So an appropriate median, 10%, and this needs to be adopted. We can't have this case where fishing for the same animal on similar grounds by similar organizations, we've got estimations of 5% and 25%. So it's time that we maybe took a national approach at thinking of what this efficiency needs to be. Thank you very much.
Thanks, Tom. That was fantastic. You packed a lot of results into that. Thank you very much indeed. We'll move on to Oscar Bos' presentation. Oscar's actually on fieldwork at the moment, so this is going to be a recorded presentation, but Pauline Kamermans is available to answer questions in the Q&A session.
Good afternoon, everyone. My name is Oscar Bos. I work at Wageningen Marine Research, and today I will tell you something about our project EcoFriend, in which we, amongst others, monitor an oyster restoration pilot in the offshore wind farm Gemini. But let me first introduce our team for our day at sea, which I will show you today. We have Joost and Mirjam from Bureau Waardenburg, Pauline, who you all know, I'm sure, Linda, also from our institute, and on board we have always a person who represents the wind farm to check whether the things we are doing offshore are, safe, and he also takes care of the communication with the wind farm, and we have the crew of the vessel. The aims of this project are, the first aim is to monitor the oyster pilot in the Gemini wind farm, and we try to, within this project, develop methods with which we can monitor oysters in a cheap way. I will tell you about that later. The third aim is to understand and model the abiotic environment, so we can predict, for example, when oysters spawn or how structures that we place are moved. If you paid attention yesterday, you could have heard that in Luca's talk, Luca van Duren. And after the project, or just before the project's finished, we will provide advice to the other projects about the things we learned. So today I'll take you a day at sea, and I will show you what kind of trade-offs and challenges you face when you want to monitor oysters far away, and possibly in a little bit different way than is usually done, which is with a small vessel, Most projects use a large vessel, which costs about 20 to 40,000 euros a day. And we try to use a small vessel, a crew transfer vessel. You can see it in the picture on top of my head. It's a kind of taxi boat, and usually they can go very fast because they have jet engines. So you can travel large distances, but the possibilities of monitoring are, of course, a bit limited because there's no crane on board. You have to do everything by hand, hoisting, etc. So we try to figure out what is possible if you use a small vessel like that. As I said, the distance to the wind farm is about 80 kilometers one way. So that really reduces the number of vessels that you can use in the first place if you want to do it in a day trip. And this type of vessel doesn't have any beds. And last year, during the Corona crisis, we were not even allowed to do multi-day trips. So actually a day trip like this was quite good for the sampling. Another consideration is how many stations do you want to do, and does it fit within your 1 day of sampling? And we think with this kind of small vessel, a very fast vessel, you can actually do quite a lot of things. Then the second question is, when you're planning this day, when do we go in the first place? So what you want is a large window in which you can operate at the wind farm site. So the first date that you focus on is the dates around neap tide, when the low tide and high tide are closest in terms of water height, water level. You can see it in the first graph. So in this case, the 3rd of August, the 19th of August, those are the dates around which I'll plan the monitoring. Then on that date, you want to have a window at a certain location during which you can sample. You have to figure that out as well. For that, we use a program that we use in scuba diving, actually, at the North Sea. And for every position on the North Sea, the model can predict how long of a dive window you have with not too much current, so you can easily do your sampling. And the third thing is, once you have got those two things, that of course the weather has to be good. So there should not be too much wind. The wave height should be typically less than 1.2 meters, more or less. Otherwise, sampling becomes really difficult, especially, for example, if you sample eDNA, you can get really sick of doing that if the waves are too high. So we love Windfinder. It's one of a good program. And actually, the wind farm has a weather station in the wind farm, so you can look around that weather station whether the weather is good. Then, a small vessel, what can you do and what is not possible? So in the first place, everything has to be quite light, because you cannot lift anything heavier than 25 kilos, according to the law. And in a wind farm, there's restrictions with regards to finding back things. So for example, you cannot place a block of concrete with a buoy on top, because you're not allowed to place anything. So you have to be able to put, so you have to put things down which are stable, which have a proven track record of stability. You can attach acoustic devices to them to plop up when you call them, but you cannot use buoys. So that's a bit of a difficult thing. And so losing equipment, so for example, we placed cages with oysters. Losing them is really not an option. And Luca talked about that yesterday. Another thing is, if you're really far away, you have to have all the spare parts, all the tools on board in case something goes wrong. And a lot of times equipment is there, but it's too heavy or it's not suitable for use in a wind farm. So Joost, I showed him in the beginning, is trying to develop tools that can help us doing things in a lightweight way. Like, for example, developing a cage that can release oysters, or kind of oyster station where you can place oysters with help of an ROV. This is a small ROV. I wanted to show that too. So we are now in the process of talking with the wind farm that we can use our small ROV to do transects. But for that, you need a lot of paperwork. Now, that's the next thing I will show. So whenever you work in a wind farm environment, you have to know that they have very high standards of safety and awareness. And so, for example, if you want to work there, you have to do a survival at sea course, which takes a few days. You have to do an exam that you understand the wind farm. So that's a YouTube movie with an exam in the end. You have to have medical approval that you're a healthy person. The whole ship and the whole every member on the ship, the crew members, they have to be in the operation control center, which is kind of the traffic center of the offshore wind farm. They have to be in the system, and you have to have a good relation with the person who is responsible for the ecological monitoring in the wind farm. That's typically someone who works for the wind farm and who is a biologist or understands what we want, and he will talk to the technical people to see what's possible. So high safety standards, really difficult to do anything at all, actually. So once you know that, it's logical that you will have to prepare a plan for every trip that you do. It's called a risk assessment and method statement. So you look at all the risks of all the methods. For example, can the wind farm or can a tube be entangled? Can something be damaged? Can something be dropped overboard, which you don't want? And all of that is assessed. The risk is assessed, and you describe that risk, and you try to describe as well how to prevent it. So for every trip, you do that. And then when that plan is ready, we have permission, we'll get permission of the Operation Control Center to enter the area. So for every trip, you need permission of this traffic center. And the traffic center actually looks whether there's also, for example, other ships that do maintenance on the wind farm. So there should not be any spatial problems. And as I said, to control all of that, we have somebody representing the wind farm to check us. And usually it's a very nice guy who likes to help as well. But that's how it works in the wind farm, basically. So whenever we go, the result is the weather is always so nice when you go sampling. But as I more or less explained, that's how I plan it. And then just to show some real results after all the difficulties and trade-offs that I talked about. So what we do is, for example, we sample eDNA to look at the fish around oysters, oyster reefs. And here are 3 locations: the Gemini Wind Park in the north, Borkum Reef along that transect in the south, more towards the coast, and then station in the middle. And you see typical sand related fish species, for example, sand eel, they are there, but also one stingray was detected, which was interesting. And then, so we have the fish community, and of course we have 2 locations with oysters, Gemini Wind Farm, as I explained before, and the Borkum Reef. And we check both locations 3 times per year for the presence of larvae near the seafloor. So we put a big tube down to the seafloor, which is about 20 to 30 meters deep. And we pump up the water, we sieve the water, as you can see in the picture, and then we count the larvae. And this has been presented before, so I didn't want to pay too much attention to it. But the first thing is that apparently the oyster restoration works because they reproduce, larvae are there, which is great. But the strange thing is that on the location in between the 2 restoration sites, we find most larvae, while there are supposed to be no oysters in the first place. So that's an interesting result, which we still have to model, basically, as I told in the beginning, to understand if these larvae can really drift from the source to that location, or that we oversee some kind of native location, which is a native oyster population, which is still there. So that was my talk. I hope you understand a little bit how it works if you want to work offshore. And if you have questions, my colleague Pauline is waiting for you. Unfortunately, I'm virtually here, but not in reality. And if you want to know more about our project, just Google EcoFriend in Wageningen and you will get there. Thank you very much.
Thanks, Oscar. I bet the weather's not as nice today as it was in those photographs. Anyway, we need to move on quickly again. The next speaker is Andrew Hunt. He's a fellow of the Society for Underwater Technology and a fellow of the Institute of Mechanical Engineers, working in marine renewables. And he's been sport diving for 30 years and is currently the expeditions advisor for the British Sub-Aqua Club.
Thank you very much, David. I am a volunteer for the British Sub-Aqua Club and serve on its National Diving Committee. And I earn my living as a mechanical engineer working in the field of subsea engineering and marine renewables. And it's been my diving career over 30 years as is gradually being fused with my work. And I'm trying to fuse it even more to spend more time on oysters. Today, I want to tell you a little bit about the British Sub-Aqua Club, for those of you who don't know about it, its Operation Oyster project, the achievements this year, some about the project phasing, our discoveries, the key challenges this year, and some next steps. So first up, about the British Sub-Aqua Club. It was founded in 1953. Its current president is His Royal Highness Duke of Cambridge, Prince William. It's a not-for-profit organisation, and as well as being a diver training agency, it's the governing body for the sport of diving in the UK, and has a remit to promote underwater science and exploration. And it currently has some 20,000 plus volunteer diver members. BSAC Operation Oyster is a citizen science project which started in April 2021, just as the sport diving community were emerging from a COVID winter lockdown. It's been a while since the last large-scale environmental BSAC project, which was Operation Kelp, led by the late Dr. David Bellamy way back in the 1960s. The idea for this Operation Oyster project came about from the successful Ocean Quest photography competition, which was organised by the Zoological Society of London and BSAC. And that sought photographs of native flat oysters. And it was that competition which brought the native flat oyster to my attention. And what Operation Oyster aims to do is to mobilise UK divers, snorkelers and beachcombers in helping rehabilitate the UK's native oyster populations, which is one of the UK's BAP priority species. The achievements this year, we've set up an online portal on the BSAC website to educate divers on the importance of the native flat oyster, and that includes an introductory video and an identification guide. The website also has an online form for collecting data on locations of native oyster remnant populations and including their competing species. We've promoted the project through the diving press and a recent TEDx talk, and have trialed data collection with volunteer divers. And very importantly, we've established supporting links with key stakeholders, the Blue Marine Foundation, the Zoological Society of London and the University of Portsmouth, whose help was instrumental in helping us get this project going. In terms of phasing, we're currently at the end of the initiation phase, where we've trialed some of the systems for wider rollout. We're entering the discovery phase with numerous diving and snorkeling expeditions in the planning stage for next year. Data collection is a deliverable of those expeditions, and we are working on feeding that data into the National Biological Network database for use by the wider scientific community. Ultimately, though, Operation Oyster is a restoration project, so we hope that this data will help plan future restoration projects and possibly be useful in gathering genetically diverse broodstocks for cultivating disease-resistant oysters. But that restoration side is down track when suitable project sites are identified and funding and other partners are secured. So on to discoveries. Most of the diving and most of the diving and beachcombing activity this year has been around the Isle of Wight, simply because that's where I live. However, we've received reports from the length of the UK, including recently from Orkney, where there might have been some possible shells of juvenile oysters found on the shipwrecks there, and as far south as the Isles of Scilly, where some old shells were found in quite deep water. As you can see from this chart, most of the findings of live specimens haven't really been in the Solent, which used to be a commercial fishery, but rather from just outside the Solent. We were hoping to explore the old commercial fishery site in Newtown Creek, but unfortunately we were forced to abandon diving attempts there on safety grounds due to the presence of a large quantity of yachts at the time. The thing about areas commonly associated with oyster beds is that they're generally of little interest to divers, being intertidal areas or flat areas of seabeds dredged within inches of destruction. So we're initially asking divers to spend a couple of minutes on their dives on their normal dive sites around the UK, which are usually shipwrecks or reefs. And here's some photographs of those findings so far. As you can see from these slides, oysters can be quite difficult to spot in the wild, but once divers' eyes are tuned in, that gets a bit easier. We've seen live specimens on numerous wrecks, usually larger solitary specimens, but in some places younger specimens in closer proximity. And in these examples here, you can just see how well camouflaged they are against the substrate. There is an idea of using shipwrecks as nodes for reseeding of oysters, and this is definitely one of those ideas I'd like to explore further down track. We've also found oysters in amongst beds of slipper limpets in the tidal waters around the island. We've not found too many closely clustered oysters like these specimens, again on a wreck in tidal waters. But one of the more interesting sites is a protected wreck site off the island. We found a high density of oyster shells, but interestingly no live specimens. We've also found small areas of oyster beds away from the reach of dredgers with densities of oysters around 5 per metre squared, thereby just about meeting the definition of an oyster bed. Our key challenges this year have been related to the pandemic. A longer than normal layoff period for divers raised safety concerns, and so BSAC's priority has been getting sports divers back into the water safely. Clubs and centres have also been focused on a backlog of diver training, boosted by an increased demand for divers wanting to learn at home. Volunteer time, being a volunteer organisation, is usually a challenge, especially to try and find a resource in a timely manner when you need it for the project. However, the response so far has been excellent, including from the younger generation of environmentally aware divers. BSAC Operation Oyster is in part a response to the demands of the wider BSAC membership wanting meaningful expeditions so that they can go diving with a purpose. The priorities for 2022 are diving and data. And we want to discover remnant and historic populations around the UK. A number of diving expeditions are to be announced soon, and these will be open to suitably qualified sports divers. And we're also continuing to explore opportunities with other groups and bodies to help restore native oyster populations and evolve the project accordingly. And what I'm very pleased to announce is that We're going to be working together with Seawalding on some e-learning and snorkel surveyor courses to help promulgate that restoration message and mobilize a citizen science army / navy. So please do feel free to contact me on the emails on this slide, and thank you for your time and listening. Thank you.
Thank you very much for everybody's contributions today, the presentations, and for your engagement in the questions as well, please. That was really super. So much work still to be done. It always has struck me that we have got a pretty good evidence base for native oysters, but as everything, it's not complete, and there's an awful lot of work still to do, and the amount of survey effort that's been put in tends to be located in sort of specific places, and building that evidence base is a sort of never-ending task. And as I was mentioning in my presentation this morning, that constant cycle of monitoring and feedback into what we're doing is so vital. So the handbook is a tremendous contribution to keeping going with that. Thanks very much indeed.
So the first of our official talks from our working group chair people is from Ruth Thurstan. Ruth Thurstan is a senior lecturer at the University of Exeter. She's working to understand the marine social ecological dynamics over the last 2 centuries using a range of historical sources. She's chair of our Historical Ecology Working Group, which is an interdisciplinary group consisting of academics and practitioners from across Europe. And we're very excited to see the first preview of the efforts that have been ongoing for some time now. Thank you, Ruth.
Thanks so much, Philine. I hope everyone can see the slides and hear me okay. And I'm really excited for the opportunity today to update you on the work that's being conducted by the Historical Ecology Working Group membership. So we have members from a range of countries, and I'll start by saying, if you see your country is not on this list and you know of historical native oyster records, please do get in touch with me. And I also want to acknowledge the large number of collaborators within this working group, and this is not an exhaustive list of everyone in the working group, but these are many of the people who've contributed to the data that I will be showing today. And again, I hope I have got everybody in here. I will, however, say that any errors are solely mine, but so it's been really wonderful to work with everybody. So the aim of the Historical Ecology Working Group is to assess the historical distribution and trends in native oyster habitat and its fisheries through time. So historical can mean a lot of different things to different people. We have some data from collaborators going back thousands of years, but in general, this working group is looking back sort of decades to centuries into the past, and mainly a lot of our data that we're collating is from the 1700s onwards, because this period corresponds with a rapid rise in the exploitation of oysters, and also documentary sources becoming more prevalent at that time. So many people will be familiar with this 19th century chart of the North Sea and coastal areas. In fact, it's been shown a few times already in this conference, and I put it there because it was published at the point in the 1880s when many oyster habitats were already severely degraded. There were some new ones being discovered, but many were very, very degraded by this point. It's also limited in its resolution and its geographic range, of course. So we're trying to really provide sort of information to build on figures such as this one, for example. And so some of the information that we are currently gathering are things like the characteristics of historical oyster habitats. What did likely or even unfished oyster habitats look like, if we can find that? What species are associated with them? What scales did these habitats exist? We're also looking at the locations of these habitats, either through fisheries or unfished habitats, and their significance, hopefully kind of trying to understand how many oysters were on these habitats through landings trends, price trends, employment, number of boats, that sort of thing. And also, although this is not developed as much yet, but something that is of great personal interest to me is cultural associations. You know, what sort of cultural memory continues to exist, and where are the places where it used to exist but has been lost? Because again, that has been mentioned by speakers such as Betsy Peabody, in terms of the community and the culture is incredibly important to these restoration efforts. So the sources that we're going to be using include, or that we are using at the moment, include government archives, newspapers, popular literature, peer-reviewed scientific and humanities literature, but also museum collections, nautical charts and surveys. Those are some of the examples. And what I'm not going to be able to do today is to give you a complete picture of historical habitats and extractive patterns. It's still a work in progress, but what I can demonstrate to you is the type and quality of evidence that we're uncovering, and I can show you where we've got to in this rather large undertaking. So first of all, habitat characteristics. In general, first-hand descriptions of oyster habitat historically are very rare. Still rarer are descriptions of habitat that hadn't been fished in some form, but we do get some indications, and I think these echo what restoration projects are finding today, particularly from some of the talks that I've been listening to today and yesterday. One of the most frequently used terms in the historical literature, and at least in the English language, is the use of clumps or lumps of oysters. So this is from Strangford Lough in Northern Ireland in the early 1600s, heavy laden beds with oysters in clumps. The same in the North Sea, people would talk about heavy clumps, which concerned trawlermen at the time, in the period before steam trawling. This is in sort of Aberystwyth Bay in the Irish Sea. There are oysters in patches. They are in lumps together. And even, so this is also in the Channel, and they talked about clumps, and this was at the period of the very end of a fishery, really within sort of the Plymouth region, but they were still finding them in clumps at that point. You also get the odd sort of tantalizing glimpse of, oyster banks of Wicklow have become hard like a rock, and it would do the banks great good to be broken up. So, I'm guessing this meant that oysters had settled too densely to be easily dredged up by the less weighty oyster dredgers or by hand, but it's an open interpretation, and I welcome people's thoughts on this. Habitat could also extend over wide areas, as we saw from Bernadette's talk yesterday. And from this description from the 1700s of a bed extending over 2 miles in length and near 2 in depth, or breadth, we might call it these days, and starting about 1.5 miles from the shore. Now, descriptions of beds this size and far larger were common, such as this second quote, nearly 80 miles long and 25 miles wide. It's rough ground, and to the point where the oysters were numerous enough that trawlers were quite concerned about getting in amongst them. And I doubt that this was contiguous habitat, but it certainly suggests that the oysters were really quite numerous over quite wide areas, and densely enough that some trawl vessels would avoid them. And I also added this in because there was a talk today talking about oysters being at low tide. And that was really interesting because we found a handful of quotes about that as well. At spring tides, the oysters are left by the sea, or where people go to gather them by hand and they go and pick them up. So that's in a few locations as well. We also have some glimpses of associated species, although I think the restoration projects are going to really give us a very good insight into this. But, you know, back in the day, people would talk about tree-like corallines, they talk about young oysters on top of those, they talk about sponges, they talk about Sabellaria habitat, they talk about ascidians and hydrozoa, and even other small shellfish. I haven't found anything that any historical evidence about eels as yet for those who were in yesterday's sessions, but it would be very exciting if anyone did have that. So we're gathering some really rich qualitative information, but we're also looking for data to understand when oyster fisheries declined and by how quickly across different regions. And apologies, this is very UK centric at the moment. I've got a lot of data from other places, but still pulling it together. And I'll just show you a handful of studies for now, but so it's just to show that a lot of information is qualitative. such as this quote from the Isle of Man, where they said it took 20 boats 7 years to dredge away the oysters. They were thick before that. But also we have sort of more quantitative information where we can get these trends through time. And so this example includes Newhaven in the Firth of Forth in Scotland. So they recorded annual landings of up to 30 million oysters in the very early 1800s, and their fishery collapsed just a few decades later. I've got an example here of the Jersey fishery, and they had higher landings again. Between 50 and 60 million oysters in 1 year was recorded. Of course, these are all estimates, so it's difficult to know whether these are overestimates or underestimates. It depends on the specific source and where they've collected the data from. And of course, and then there's Swansea, which is in Wales, where they recorded 9 million oysters. And then it again declined really quickly. All of these, you see this very swift decline, but in Swansea, it persisted into the early 20th century. In all of these, a red line indicates when annual data started to be collected by fisheries boards or the like. And you can see that those start a long time after these oyster fisheries at least have declined. And so we're gathering these from a range of earlier sources, some of them governmental data, some of them more popular literature or newspapers. I strongly suspect that these peaks that we can see, so for example, in these fisheries, were sort of, these early peaks were very short-lived, and that prior catches were substantially lower, but this is something we're working on finding out. We're also mapping the locations where oyster habitat and fisheries were recorded in the past. Now, as you might expect, descriptions of locations span varying degrees of precision, ranging from extremely vague, such as this top quote, all parts of the channel, to highly precise, so the naming of distinct fishing grounds within relatively small geographical areas or bays, for example. So getting on to this is the data that we have recorded from the 19th century so far. Now, the data are mainly based on references to fisheries. There's also some historical sort of scientific survey data included. We haven't quite finished updating that yet. Very notable that the fisheries records may also not be complete. Well, I know they're not complete, so do let us know if you have any records for areas where there's no blue dots. This is for the 20th century, so for the 1900s, and this looks a little different due to two factors. Firstly, there was a very real decline in oyster, well, sort of commercial oyster fisheries in some locations. So say, for example, along the east coast of Scotland, along various parts of the English and Welsh coast, and also parts of the Irish coast as well. But also there's more comprehensive record keeping in later years, possibly also influenced by the more recent literature being more accessible or more well known by researchers as well. So this is also, again, something that we're working on, but it's just to say it's not perfect at the moment, but we're making progress. And we're working towards building this map of historical extent. So this map is informed by data from the 17th to the 20th centuries. And I would like to acknowledge Hannah McCormick at ZSL for the GIS figures. She very kindly got those to us at short notice. And again, if you know if there's additional data to inform this range map, please do let us know. And that's it from me. That was a really short, sort of swift run through some of the things that we're doing. And I wish to thank again collaborators in the working group, and please direct any questions to me, and thank you for listening.
Thank you very much indeed, Ruth. And we will be taking questions again at the end of this session. So feel free to put them in the chat box, but even better if you can give them yourself at the end, because it makes it all a bit more real. The next speaker, I'd like to invite Anna Hughes. Anna has just completed her master's in marine systems and policies at the University of Edinburgh. And as her master's research project, she's led the process of collaboratively developing a site selection checklist with the Site Selection Working Group here in NORA. Thank you, Anna.
Thanks, Philine, and thank you so much for everyone who's attending this session today. And a special thanks to the NORA Organising Committee for doing such an incredible job on this online conference. So my name's Anna Hughes, and I'm delighted to be introducing the Native Oyster Habitat Restoration Site Selection Checklist today. This has been an ongoing piece of work involving many members of the NORA Site Selection Working Group over the past year. And it all began, as Philine mentioned, as my master's dissertation, with Philine as my supervisor, and it's grown from there. This research has looked to determine the most important factors in site selection for the European native oyster habitat restoration projects. Ooh, I am struggling to put my slides forward.
Sometimes you have to activate the screen by clicking on it first and then using the spacebar.
Oh, there we go. Thank you. So my project aims, the main aim of this project was to draw upon pan-European expertise in order to reach consensus on the most important factors in determining site selection for European native oyster habitat restoration projects. Site selection has been determined as a priority research area, which led to the dedication of the Site Selection Working Group. Since site selection is such an early step in the restoration timeline, the execution of all sequential stages depends on effective and appropriate site selection. We wanted to create a synthesis of current knowledge on site selection and produce resources that were useful to restoration practitioners. So there were three main outputs from this project. The first one was my dissertation, which I completed back in August. The second one, and the one that will be the focus of this talk today, was the site selection checklist. So myself and Philine wanted to adapt my thesis into something that would be more useful in the field, which is where this site selection checklist comes in. This document is a sort of short and snappy summary of the most important points of my master's dissertation and aims to provide foundational and practical site selection advice to restoration practitioners working in the field. And then the third output, which I'll switch my focus to after this conference, is the peer-reviewed publication. So before I introduce the checklist itself, I'm just going to quickly go over the methodology that we've used in this work. So we used the Delphi technique to gather our information, which is an iterative and anonymous questionnaire method. It's a form of expert consultation, which is really fantastic for bringing together large groups remotely and has been shown to deliver excellent results when trying to reach consensus and distill complex topics. The greatest wealth of knowledge in site selection is currently held by this network of practitioners and scientists across Europe, and it's not necessarily in all cases published into articles yet, so we felt that the Delphi process would be perfect to tap into this expert knowledge base. We conducted 3 rounds of questionnaires between autumn 2020 and spring 2021. And alongside these questionnaires, I also conducted a literature review, and we held working group meetings to further discuss the questionnaires and fill any gaps in our understanding. These discussions were incredibly helpful in contextualizing the questionnaire responses and also allowed all of our participants to share their thoughts and ideas. I should add that the Delphi technique requires anonymity for valid results, so all of these discussions were held after the completion of the 3 rounds of questionnaires. So although they didn't change the actual results, they were really important in adding context into the checklist. And resulting from this process, we came up with 65 factors that are important to consider in site selection for European native oyster habitat restoration projects. These were then split into essential and desirable for site selection. This allowed some prioritization and hopefully makes this big list of 65 factors a bit less overwhelming. We also split them into these 5 subsections. So we have threats to native oyster habitat restoration, project logistics, and then abiotic, biotic, and socioeconomic factors. That should be enough background to move on and talk about the checklist itself. So here we have our front cover. So as you can see, it fits really nicely with the other NORA handbooks, which Philine was speaking about earlier. This is the first time that the expert contributors have seen it, so hopefully everyone is as excited as I was. I'm incredibly pleased that we were able to produce this checklist, which I hope will transform the research which started as my dissertation into something really practically useful in the field. So we have our contents page just here. This sort of just shows you the structure of how the document is put together. It's similar in format, but shorter to the other NORA publications. It was authored by myself and Philine, but we absolutely could not have done it without our 26 expert contributors, whose names I'll leave up on the final slide. So for the sake of time, I'm not going to go through the whole document, but it will be available for you to look at tomorrow in the projects and partners section of GatherTown. It'll also be very shortly going up on the NORA website. So this is just a screenshot, just a section of the checklist itself. Altogether, the document's about 20 pages, but this checklist in the center is very much the focus of it. So there are these 65 factors which are important to consider for native oyster habitat restoration. And we intended practitioners to use this a bit like a risk assessment. So all factors on the list should be considered, all factors on the list should be considered. However, the inability to meet one or several of the factors, particularly those we've listed as desirable, shouldn't undermine the restoration efforts. And although this list is universally applicable, it should be adapted at the project level. Just for usability, we've color-coded this checklist into essential and desirable, and we've again broken it down into the 5 subsections. So here I'm just showing the section for abiotic factors. Another part of the checklist which I'd like to show you today is this wonderful infographic that we had created. I'm really pleased with how it came out, and this is showing the essential factors the essential factors to consider in site selection. So each of the circles represents a different essential factor, and the most important of these are marked with the little red exclamation mark. These exclamation marks are the factors that we felt either pose a direct threat to oyster survival or would completely prohibit project establishment. So in other words, these are the absolutely critical factors that no project is likely to succeed without. And the color of the circles represents again these 5 subsections, which are just shown in the bottom left here. So please do go and take a look at this once it's available in the projects and partners and on the NORA website. The remainder of the checklist document provides a brief introduction to the importance of each factor on the checklist, as well as indicating which locations and contexts factors may be of particular relevance to. So this is to provide some in-field context, and again, it's based on a compilation of expert knowledge from the questionnaire responses and further discussions. We've also included tolerance ranges and thresholds where appropriate throughout the document. And lastly, we've also added these little icons on the right here throughout this section where we felt like the use of indicator species or observation of the benthic community or reference to locally relevant legislation would be useful to practitioners rather than direct measurement in some cases. Again, this is site specific, but we've just added these icons as like a little helping hand where we think this might be helpful. And the checklist document comes to an end with some references which we included throughout where we felt that further reading might be useful to restoration practitioners. And that wraps up the checklist and also wraps up my presentation for today. As I mentioned, it will very soon be available to download from the website and in the projects and partners room. The next step for me is to start drafting the peer-reviewed publication, and I'll be available to take questions at the end of this session, or you can email them to myself and Philine. And then lastly, just a huge thanks to everyone who was involved. We had 26 expert contributors altogether, and they all provided fantastic insight. So we really couldn't have done it without all those amazing contributions. Thank you.
Thank you, Anna. Much appreciated. I'll move straight on to the next presentation, which I'm giving on behalf of Åsa Strand, who is unfortunately able to join us today, but Åsa has been pivotal in moving in coordinating up to date our production working group, although, as I will talk about, this is, sorry, let me start again. Hello. My name is Ermgassen, and today I am presenting this on behalf of Åsa Strand, who is unable to be with us today. This is a summary of the work that's been ongoing by the production working group over the last couple of years. which over 30 people have attended various workshops for, etc. So what have we actually been up to for the last couple of years? Well, we had a workshop last December following on from the NORA 3 conference, another one in September following up on that and building further on the discussions that we initiated. Bérenger Colsoul from AVI has launched some informal knowledge exchange meetings, and those will be continuing. So if you are interested in having informal conversations about how to produce oysters and any troubleshooting methods, etc., please do get in touch with Bérenger and he will add you to the list to make sure you can join future calls. And we've been developing information sheets for both oyster producers and restoration practitioners, and also in the process of developing a policy brief and prospective note for the aquatic living resources special issue. So please do bear in mind that prospective notes are also an option if you're considering submitting anything to that special issue. So our workshop last December took place online, and we had over 30 contributors. It was a really engaging conversation to be had between oyster producers and native oyster restoration practitioners. And through that discussion, it became clear there was an identified need for an increased understanding of one another's industries. And so as a result of that, we opted to devise, to construct text for 2 information working sheets that I will introduce you to today. The second thing that became apparent is that there are really major barriers to increasing oyster production from existing producers as a result of the mismatch in the timelines between commonly available restoration funding and oyster restoration oyster production. And so that's what the topic that the policy brief and the aquatic living resources prospective note will be focusing on. So the 2 information sheets are produced essentially with different perspectives. One is from the point of view of oyster producers, oyster practitioners, to inform oyster producers what they might need to know if they're thinking about engaging with oyster restoration projects. And then there was one from the perspective of the oyster production industry about information that they felt they were commonly answering questions on from oyster restoration practitioners. And as we've already seen, there's an increasing number of projects and increasingly these conversations are happening. So we're hoping that these information sheets can essentially move that conversation forward so that people already know what questions they want to ask when they are addressing each other, so to speak. So zooming into the oyster producers, what oyster producers need to know about oyster habitat restoration. This was divided into 4 topics. What are oyster habitat restoration projects trying to achieve? And this was summarized using the fantastic infographic from the UK Ireland Oyster Restoration Handbook by Preston et al., outlining the ecosystem services and the biodiversity value of reefs. The second topic was what it takes to manage an oyster habitat, native oyster habitat restoration project. And for that, again, we leaned on the handbook and borrowed this infographic showing the timelines. And then we wanted to summarize, the restoration practitioners wanted to summarize what kind of oysters they're looking for. And this was summarized as essentially wanting to make clear that they're not looking for an oyster that looks or tastes good, but there are other requirements that differ from that of the table market, which is, for example, there has to be certainty that the seed aren't mixed with dry gas seed, that biosecurity in terms of epifauna and disease there's also a high level of information and certainty about those. And also that the product might be quite different in terms of spat-on-shell. So it's something we've already heard again a little bit in the conference, but there is a strong interest in having spat-on-shell as opposed to singles because of the potential predation refuge that might provide. And the other thing is there might be a stronger emphasis on broodstock genetics or concern about effective breeding size in terms of using those as a source population for a restoration project. And the final point that a lot of restoration practitioners felt was important to make is that it was important that oyster restoration producers were able to deliver on orders because they have project deliverables essentially that are dependent on that. So they would like to be a valued customer. So moving to the second worksheet, what restoration practitioners need to know about the oyster production industry. In this one, we focused on outlining and giving a brief overview of the 3 primary production types for seed, and then talking about the reliability of supplies and making sure that there's a common understanding, also about timelines of production, how understanding there might be costs associated with genetic and biosecurity requirements as they differ from standard procedures, or may differ from standard procedures. Thinking about grow out, and then also understanding a little bit better, what the traditional market is, so that there's no misunderstandings. So in terms of the 3 primary production types, we gave an overview of seed-based collectors, pond-based production, and hatchery production. And thanks to Bérenger Colsoul for sharing his fantastic figure here. You will also be able to see this figure if you visit the RV booths in the project area and you visit their website. We also worked with everyone in the team to pull together this, hopefully, what I find incredibly useful timeline of oyster seed production. It looks at what the timing for various activities are for all 3 of those production types across different geographies or between different hatcheries. And hopefully this provides then an understanding for oyster restoration practitioners about what the lead times are and when they need to really start thinking about approaching producers if they want a product, say, in the fall. And then thinking about beyond oyster seed and grow out, we work together to pull together data, growth data over time from a range of different geographies, so that we could explain then also how growth rates differ within the different ranges. So again, the expectation is that practitioners have a sounder understanding of how long they might need to wait to get an oyster to a certain size in their geography. And also an understanding of the potential for quite steep declines in survivorship following relaying. And the bottom figure is based on data from IVL in Sweden, and it's looking at different culture types, so suspended culture against bottom culture, and also different starting sizes of oysters. And again, we're hoping that this information at your fingertips can help restoration practitioners understand better what kind of product might suit their projects best. And the main considerations they wanted to that the oyster producers highlight in the information sheet is that there needs to be an understanding from practitioners, restoration practitioners, that it is essentially a bit of a risk in terms of there's some risk because it's new and there's investment required. So even though there is a financial reward, overcoming these issues with regards to timelines is going to be very important in terms of growing these production relationships into the future, and understanding that these adaptations also come with a cost. So thinking about engaging with oyster producers early on in the project, possibly even at the grant writing stage, so that those costs can be accounted for, could be really important. And those are the kinds of mismatches, as you've noticed, between the 2 information sheets. There is a discourse that we will be exploring further in this policy brief and perspective note to try and really hammer home the fact that longer term funding is really going to be critical for allowing oyster restoration projects to grow if they need seed and they need increased oyster supply. And with that, I'd just like to finish by thanking all of the participants, but listed here are those who've co-authored those information sheets as well. If you're interested in participating in the policy brief or the prospective note, or engaging in those informal calls, please do get in touch with Åsa, Bérenger, or myself, and we will make sure you are appropriately connected. So thank you very much for listening. Oh, and also those information sheets are now available for download in the project zone as well. You'll find there's a booth for them. So please do go take a look. I hope you find them very useful.
As maybe I need not remind you of the fact that the happy hour will now start in Gathertown. When you leave the Zoom, you will go back to Gathertown. And of course, the great attraction will be that for the benefit of the public present, Mr. Luc Helmer's avatar will jump over men and horses, hoops and garters, and maybe through a hogshead of real fire. After, at the end of, well, I think a pretty full and maybe tiring day, I find it fantastic that so many people took the time to do this, and the great comments we received. Thank you all very much. Well, for the whole day was fantastic. See you maybe see you maybe in Gathertown, and otherwise this night, and otherwise tomorrow morning. Bye-bye.